Hello, and welcome to this quiet place where science and stillness meet. Tonight, we're going to step beneath the soft canopy of cherry blossoms, those delicate flowers that arrive each spring with a whisper of pink and white, carrying with them one of nature's most gentle lessons about renewal, impermanence, and the quiet rhythms that govern all living things. There is something about these blossoms that invites us to slow down, to notice, to breathe a little more deeply. You can let your shoulders settle if they haven't already. Your breathing can find its own rhythm, steady and unhurried. Perhaps you've had a long day or a series of them. Perhaps your mind has been moving quickly, sorting through tasks and thoughts and the endless small details that fill our waking hours. Here, in this space, there is no urgency. The cherry blossoms have been blooming for millions of years, and they will continue long after tonight. Their story is patient, and so are we. If these journeys bring you comfort, you're welcome to return anytime. There are many quiet paths through science, and each one offers its own kind of rest. Let us begin, then, with the blossoms themselves and the remarkable facts that make them not just beautiful, but deeply, quietly fascinating. Cherry blossoms belong to the genus Prunus, a group of trees and shrubs that also includes plums, peaches, apricots, and almonds. Within this genus, there are hundreds of species, but when we speak of cherry blossoms in the way that stirs something in the human heart, we are often speaking of a smaller group, particularly those in the subgenus Araceus. These are the true cherries, and among them, the most celebrated for their flowers are the Japanese flowering cherries, varieties that have been cultivated not for fruit, but purely for the beauty of their blooms. It is a rare thing in human history to nurture a plant solely for the way it makes us feel when we look at it. The most iconic of these is Prunus cerulata, the Japanese cherry, though even within this single species there exist dozens of cultivated varieties, each with its own subtle character. Some have petals that open wide and flat, five-petaled and simple. Others have been bred over centuries to produce blossoms with dozens of petals, clustered so densely they resemble soft pom-poms or delicate roses. The colors range from the purest white to pale pink, and in some varieties, a deeper rose that seems to hold the last light of sunset. Each variety blooms at a slightly different time, extending the season of blossoms across several weeks, a phenomenon that the Japanese call the sakura zensen, the cherry blossom front, a wave of flowering that moves across the islands like a gentle tide. The blossoms themselves are ephemeral. Most cherry flowers last only a few days once they open fully. Some varieties hold their petals for a week, but many release them after just 3 or 4 days, especially if rain or wind arrives. This brevity is not a flaw in their design, but rather an essential part of their strategy. The tree invests enormous energy into producing these flowers, thousands of them covering every branch, and it cannot sustain them for long. The goal is pollination, and once that is achieved, or once the window of opportunity is passed, the petals fall. This impermanence has become central to the cultural meaning of cherry blossoms, particularly in Japan, where the concept of mono no aware, the gentle sadness of transience, is deeply woven into the appreciation of sakura. But before we drift too far into philosophy, let us stay with the science, for it is in the details of biology that we find the true wonder. Each cherry blossom begins as a bud, formed the previous summer. This is a fact that surprises many people. When you see cherry blossoms in April, you are looking at structures that began their development nearly a year earlier. After the tree has flowered in spring and its leaves have unfurled, it begins the quiet work of preparing for the next year. By midsummer, tiny flower buds have already differentiated within the branch, hidden beneath protective scales. These buds enter a state of dormancy as autumn arrives, a kind of suspended animation that allows them to survive the cold months ahead. Dormancy is not simply waiting. It is an active physiological state governed by hormones and environmental cues. The buds require a period of cold, a certain number of hours below a specific temperature threshold, before they can break dormancy and resume development. This requirement, called chilling accumulation, ensures that the buds do not open prematurely during a warm spell in winter, which would expose the delicate flowers to lethal frost. Different cherry species and varieties have different chilling requirements, adapted to the climates in which they evolved. This is why cherry trees that thrive in Japan or Korea may struggle in warmer regions, and why breeders have worked to develop low-chill varieties for places that do not experience prolonged winter cold. Once the chilling requirement has been met, the buds become responsive to warmth. As spring temperatures rise, metabolic activity within the bud increases. Cells begin to divide and expand. The protective scales that have shielded the developing flower through winter start to loosen. Pigments are synthesized, giving the emerging petals their color. Sugars stored in the tree's roots and branches are mobilized and transported to the buds, fueling this rapid growth. In the span of just a few weeks, a hard, brown bud transforms into a cluster of soft, delicate flowers. The timing of this transformation is exquisitely sensitive to temperature. Cherry blossoms are one of the most reliable biological indicators of climate, so consistent in their response to warmth that records of their blooming dates have been kept in Japan for over 1,000 years. In Kyoto, historical documents record the date of peak cherry blossom viewing, or hanami, going back to the year 812 CE. This is one of the longest continuous phenological records in existence, a dataset spanning more than 12 centuries. Scientists have used these records to reconstruct spring temperatures in Japan across that entire period, revealing fluctuations in climate long before modern instruments existed. The blossoms, in their quiet way, have been keeping a record of the Earth's rhythms. In recent decades, that record has shown a clear trend. Cherry blossoms in Kyoto are now blooming earlier than at almost any point in the past 1,200 years. The peak bloom date has shifted earlier by about a week compared to the long-term average, and in 2021, the blossoms reached their peak on March 26th, the earliest date ever recorded. This shift is consistent with rising spring temperatures across the northern hemisphere, a fingerprint of climate change written in petals and pollen. But let us not dwell and worry. Tonight is for calm observation, for understanding, for the gentle appreciation of how things work. The cherry trees are responding to their environment, as they always have, with sensitivity and precision. They are not aware of human concerns, only of temperature and light, the ancient cues that have guided them for millions of years. The structure of a cherry blossom is simple and elegant. Most wild cherry species have 5 petals, 5 sepals, and numerous stamens arranged in a radial pattern around a central pistil. The petals are the most conspicuous part, broad and often slightly notched at the tip, giving them a heart-like shape. They are delicate, almost translucent in some varieties, with a texture like the finest tissue paper. When sunlight passes through them, they seem to glow from within, a soft luminescence that is part of their magic. The color of the petals comes from pigments called anthocyanins, the same compounds that give red and purple hues to many fruits, flowers, and autumn leaves. In cherry blossoms, the concentration and type of anthocyanins determine whether the petals appear white, pale pink, or deeper rose. White flower varieties have very low levels of anthocyanins, and their petals reflect light across the entire visible spectrum, appearing white to our eyes. Pink varieties accumulate more anthocyanins, particularly in the outer layers of the petal cells. The intensity of the pink often depends on environmental factors as well, including temperature and sunlight. Cooler temperatures during bud development tend to enhance anthocyanin production, resulting in deeper pink hues. Beneath the petals, the sepals form a protective cup, green, or sometimes tinged with red. These are the structures that enclosed and protected the flower while it was still a bud. Once the flower opens, the sepals fold back, revealing the reproductive organs. The stamens, typically numbering between 20 and 50 depending on the species, radiate outward from the center. Each stamen consists of a slender filament topped with an anther, a small sac that produces pollen. The anthers are usually yellow or cream-colored, providing a gentle contrast to the pink or white petals. At the very center of the flower is the pistil, the female reproductive structure. It consists of an ovary at the base, which contains a single ovule, a style that extends upward, and a stigma at the tip, which receives pollen. In cherry blossoms, the style is often quite long, extending beyond the stamens, making the stigma easily accessible to pollinators. The stigma has a slightly sticky surface, adapted to capture pollen grains carried by insects or, in some cases, by the wind. Pollination in cherry trees is primarily accomplished by bees. Honeybees and various species of native bumblebees and solitary bees are the main visitors to cherry blossoms, drawn by the flower's subtle fragrance and the promise of nectar. The nectar is produced by a ring of tissue at the base of the flower, called the nectary, and it is relatively modest in quantity compared to some other spring flowers. Cherry blossoms are not the most generous nectar source, but they bloom in such abundance, and often so early in the season, when few other flowers are available, that they become an important food source for bees emerging from winter dormancy. When a bee lands on a cherry blossom, it typically positions itself in the center of the flower, gripping the stamens and pistil as it probes for nectar. In doing so, its body brushes against the anthers, picking up pollen grains. As the bee moves to the next flower, some of this pollen is transferred to the stigma, achieving pollination. If conditions are favorable, a pollen grain will germinate on the stigma, sending a pollen tube down through the style to the ovary, where fertilization occurs. The ovule develops into a seed, and the ovary swells to become the fruit, a cherry. However, in many ornamental cherry varieties, especially those with double flowers, fruit production is rare or absent. Double flower varieties have been bred to produce extra petals, often at the expense of functional stamens and pistils. The additional petals are actually modified stamens or other floral parts, transformed through selective breeding into petal-like structures. These flowers are often sterile, unable to produce seeds. This does not trouble the tree, because ornamental cherries are propagated by grafting, not by seed. A cutting from a desirable variety is joined to the rootstock of a hardy cherry species, and the two grow together as one tree, preserving the exact characteristics of the ornamental variety. The fragrance of cherry blossoms is subtle, not the heavy perfume of roses or lilies, but a delicate, slightly sweet scent with hints of almond and vanilla. The scent comes from a complex mixture of volatile organic compounds released by the petals, including benzaldehyde, which has an almond-like aroma, and various esters and alcohols that contribute fruity and floral notes. The fragrance is most noticeable on warm, still days, when the volatile compounds linger in the air around the tree. It is a scent that does not announce itself loudly, but rather invites you to come closer, to pause and breathe it in. Cherry trees belong to the family Rosaceae, a large and diverse family that includes not only stone fruits like cherries, plums, and peaches, but also apples, pears, strawberries, raspberries, and of course, roses. This family is characterized by flowers with 5 petals and numerous stamens, a pattern you can see repeated across thousands of species. The Rosaceae family is ancient, with fossil evidence suggesting it originated over 100 million years ago, during the Cretaceous period, when flowering plants were diversifying rapidly across the Earth. The genus Prunus itself contains more than 400 species, distributed across the Northern Hemisphere, from North America to Europe to Asia. The greatest diversity of prunus species is found in East Asia, particularly in China, which is considered the center of origin for many cherry species. From there, cherries spread and diversified, adapting to different climates and ecosystems. Some species, like Prunus avium, the wild sweet cherry, are native to Europe and Western Asia. Others, like Prunus serratina, the black cherry, are native to North America. Each species has its own ecological role, its own set of adaptations, its own relationship with the animals and insects that depend on it. The wild ancestors of ornamental cherries were small to medium-sized trees, often growing in forests or along forest edges, where they could receive ample sunlight. They produced small fruits, cherries that were eaten by birds and mammals, which dispersed the seeds in their droppings. This mutualistic relationship between tree and animal is ancient and fundamental. The tree provides food, and in return, the animal carries the seeds away from the parent tree, giving them a chance to germinate in a new location, free from competition with the parent and siblings. The wood of cherry trees is prized for its beauty and workability. It is fine-grained, with a warm reddish-brown color that deepens with age and exposure to light. Cherry wood is used in fine furniture, cabinetry, musical instruments, and wood turning. It is strong yet easy to carve, and it takes a polish beautifully. In Japan, cherry wood, or sakura wood, has been used for centuries in traditional crafts, including the making of woodblock prints. The blocks used to print the famous ukiyo-e images of cherry blossoms were often carved from cherry wood, a fitting symmetry. The bark of cherry trees is distinctive, smooth, and marked with horizontal lenticels, small pores that allow gas exchange between the inner tissues and the atmosphere. In many species, the bark has a reddish or bronze sheen and peels in thin, papery strips. This peeling bark is not a sign of disease, but a natural characteristic. As the tree grows and the trunk expands, the outer bark splits and curls away, revealing fresh layers beneath. The bark contains compounds called phenolics, which have antimicrobial properties and help protect the tree from infection. Cherry trees, like all trees, are engaged in a constant, quiet conversation with their environment. They sense light through photoreceptor proteins in their leaves and buds, detecting not only the intensity of light, but also its color and direction. They sense temperature through changes in membrane fluidity and enzyme activity. They sense the seasons through the changing length of day and night, a phenomenon called photoperiodism. All of these signals are integrated, allowing the tree to time its growth, flowering, and dormancy with remarkable precision. In spring, as the cherry blossoms open, the tree is performing an act of immense biological investment. Producing thousands of flowers requires a substantial portion of the tree's stored energy reserves. Each flower must be constructed, cell by cell, with petals, stamens, pistils, and all the biochemical machinery needed to attract pollinators and achieve fertilization. This investment is a gamble, a bet that conditions will be favorable, that pollinators will be available, that frost will not arrive and destroy the flowers. In most years, the gamble pays off. In some years, it does not. But the tree, in its patient, long-lived way, can afford to lose a season. It will try again next year, and the year after, for decades or even centuries. Some cherry trees are extraordinarily long-lived. In Japan, there are cherry trees that are over 1,000 years old, ancient individuals that have witnessed the rise and fall of empires, the passage of countless springs. One of the most famous is the Jindai sakura in Yamanashi Prefecture, estimated to be around 2,000 years old. Its trunk is massive and gnarled, supported by wooden crutches to hold up its heavy limbs. But each spring it still produces blossoms, a testament to the resilience and endurance of these trees. The root system of a cherry tree is extensive, spreading outward in the soil, anchoring the tree and absorbing water and nutrients. Cherry roots are relatively shallow compared to some other trees, typically concentrated in the top meter of soil. This makes them somewhat vulnerable to drought, but it also allows them to take advantage of nutrients that accumulate near the surface. The roots are colonized by beneficial fungi, forming associations called mycorrhizae. The fungi extend the reach of the root system, accessing water and nutrients that the roots alone could not reach. And in return, the tree supplies the fungi with sugars produced through photosynthesis. This partnership is ancient, dating back hundreds of millions of years, and it is essential to the health of most trees. Cherry trees also host a diverse community of other organisms. Their leaves are food for caterpillars of various moth and butterfly species. Their flowers provide nectar and pollen for bees and other insects. Their fruits are eaten by birds, squirrels, and other animals. Woodpeckers excavate cavities in the trunks, which are later used as nests by other birds and by bats. Lichens and mosses grow on the bark, especially in humid climates. A single cherry tree is not just an individual organism, but an ecosystem, a small world of interdependent lives. The leaves of cherry trees emerge shortly after the flowers, or sometimes simultaneously, depending on the species. The leaves are simple, meaning each leaf is a single blade rather than being divided into leaflets. They are typically oval or lance-shaped, with finely serrated edges and a pointed tip. The upper surface is usually dark green and somewhat glossy, while the underside is paler and may have fine hairs, especially along the veins. In autumn, cherry leaves turn shades of yellow, orange, and red before falling, though they are generally less celebrated for their autumn color than for their spring blossoms. Photosynthesis in the leaves is the engine that powers the tree. Using the energy of sunlight, the leaves convert carbon dioxide from the air and water from the soil into glucose, a simple sugar that serves as the building block for all the tree's tissues. Oxygen is released as a byproduct, a gift to the atmosphere. This process, occurring in the chloroplasts of leaf cells, is one of the most important chemical reactions on Earth, the foundation of nearly all terrestrial ecosystems. The glucose produced in the leaves is used immediately for energy or converted into other compounds. Some is used to build cellulose, the structural material of cell walls. Some is used to synthesize amino acids and proteins. Some is converted into fats and oils, and some is converted into starch and stored in the roots, the trunk, and the branches to be used later when the tree needs energy but cannot photosynthesize, such as during winter or during the intense growth of spring flowering. The vascular system of the tree, composed of xylem and phloem, transports water, nutrients, and sugars throughout the plant. Xylem carries water and dissolved minerals upward from the roots to the leaves, driven by the evaporation of water from the leaf surfaces, a process called transpiration. Phloem carries sugars and other organic compounds from the leaves to the rest of the tree, supplying the energy needed for growth and metabolism. This transport system operates continuously, a silent circulation that sustains the life of the tree. Cherry trees are deciduous, meaning they shed their leaves in autumn and remain leafless through winter. This is an adaptation to cold climates, where maintaining leaves through winter would be costly and potentially damaging. Leaves are vulnerable to freezing, and in winter, water is often locked up in frozen soil, making it unavailable to the tree. By shedding its leaves, the tree reduces water loss and avoids the risk of freeze damage. The nutrients in the leaves are partially reabsorbed before they fall, transported back into the tree to be stored and reused the following spring. The cycle of growth, flowering, fruiting, and dormancy repeats each year, a rhythm as reliable as the turning of the earth. For the tree, each year is both a repetition and a new beginning, a continuation of a life that may span centuries. For us, Watching the cherry blossoms return each spring, there is comfort in this rhythm, a reassurance that despite all the changes and uncertainties in our own lives, some things continue, some patterns hold. The cultural significance of cherry blossoms, particularly in Japan, is profound and multifaceted. The practice of hanami, or flower viewing, dates back over 1,000 years. Originally, hanami referred to the viewing of plum blossoms, which bloom earlier than cherries and were introduced to Japan from China. But by the Heian period, around the 8th to 12th centuries, cherry blossoms had become the focus of hanami, and the tradition became deeply embedded in Japanese culture. People would gather beneath the blooming trees to appreciate their beauty, to compose poetry, to share food and drink, to celebrate the arrival of spring. The timing of cherry blossoms depends on something called chilling hours, a quiet accumulation that happens while the world sleeps through winter. Each bud requires a certain number of hours below a specific temperature, usually around 7 degrees Celsius, before it can properly respond to spring warmth. This isn't just cold for cold's sake. The chilling period breaks down growth inhibitors within the bud, chemical compounds that keep the flower locked away until conditions promise survival. Without enough cold, the spring signal falls on deaf ears, and blooms arrive confused, scattered, or not at all. This is why cherry trees planted in climates too warm year-round rarely flower as they should. They wait for a winter that never comes. In Japan, meteorologists track cherry blossom forecasts with the same attention given to storm systems. The forecast, called sakura zensun, moves like a soft wave from south to north, beginning in Okinawa in January and reaching Hokkaido by May. It is based on temperature models, historical data, and observations from hundreds of reporting stations. People plan travel, gatherings, and even weddings around these predictions. The accuracy has improved over decades, refined by scientists who understand that a difference of 2 degrees over 10 days can shift peak bloom by nearly a week. It is a gentle reminder that nature operates on rhythms we can observe but never command. Cherry blossoms open in a sequence that feels almost choreographed. First come the earliest buds on the southern, sun-facing side of the tree. These receive more warmth and wake first. Then the rest follow in waves, each branch contributing its own timing based on exposure, angle, and microclimate. Within a single tree, you might find buds still tight, flowers half open, and petals already beginning to fall. This staggered blooming extends the display and ensures that if a late frost strikes, not every flower is lost. It is a form of insurance written into the tree's growth pattern, a hedge against the unpredictability of spring. The color of cherry blossoms ranges from pure white to deep pink, and this variation is not random. Pigments called anthocyanins are responsible for pink and red hues, and their concentration depends on genetics, soil pH, and even the temperature during bud development. Cooler nights before blooming tend to deepen the color, while warmer conditions produce paler flowers. White blossoms, like those of the Yoshino cherry, contain little to no anthocyanin, relying instead on the natural color of their petals' cellular structure. The way light scatters through these cells gives white petals a luminous quality, almost as if they hold their own soft glow. Some varieties shift color as they age, opening pale and darkening toward the center as the days pass. Yoshino cherry trees, the most commonly planted ornamental variety, are not grown from seed. They are clones, propagated by grafting branches onto rootstock. This means that every Yoshino cherry tree in a park or along a boulevard is genetically identical to its neighbors. They bloom at the same time, respond to temperature in the same way, and share the same vulnerabilities. This uniformity creates the spectacular synchronized displays people travel to see, but it also means the population is fragile. A disease or pest that affects one can sweep through all of them. Scientists are aware of this risk and work to preserve genetic diversity in cherry tree collections, maintaining wild varieties and hybrids as a safeguard. The petals of cherry blossoms are delicate, typically lasting only a few days once fully open. Their thinness is part of their beauty and their brevity. Each petal is composed of a few layers of cells with minimal structural reinforcement. This makes them vulnerable to wind and rain, but it also allows them to be light enough to drift gracefully when they fall. The falling petals, called hanafuki or flower blizzard in Japanese, create carpets of pink and white on the ground, floating on water, catching in hair and on clothing. This stage of the bloom is celebrated as much as the opening, a recognition that the end of something can be as moving as its beginning. Photosynthesis in cherry trees begins in earnest once the leaves emerge, usually just as the flowers fade. The flowers themselves do not photosynthesize significantly. Their role is reproduction, not energy production. The leaves are the true engines of the tree's growth, converting sunlight into sugars that fuel everything from root expansion to next year's bud formation. The timing of leaf emergence is carefully calibrated. If leaves came too early, they might be damaged by frost. If they came too late, the tree would lose precious growing time. Cherry trees have evolved to balance these risks, and the result is a spring sequence that feels seamless. Flowers first, then leaves, then the long green work of summer. Cherry trees are deciduous, shedding their leaves each autumn in preparation for winter. This annual cycle of growth and rest is itself a form of renewal. As leaves age, the tree withdraws valuable nutrients from them, pulling nitrogen, phosphorus, and other elements back into the branches and roots. What remains in the leaf is mostly cellulose and waste products. The leaf changes color as chlorophyll breaks down, revealing yellows, oranges, and reds that were present all along but masked by green. Then the tree forms a specialized layer of cells at the base of each leaf stem, called the abscission zone, which seals off the leaf and allows it to fall. This process conserves water and energy during the cold months when growth is impossible. Beneath the soil, cherry tree roots extend far beyond the canopy, sometimes twice the width of the branches above. These roots are not just anchors. They are explorers, seeking water, nutrients, and stability. Fine root hairs, invisible to the naked eye, increase the surface area for absorption by orders of magnitude. In one study, researchers calculated that a single mature tree might have millions of root tips, creating a hidden network as complex as the branches above. These roots also form partnerships with fungi, trading sugars made by photosynthesis for minerals the fungi extract from soil. This relationship, called mycorrhizal symbiosis, is ancient and essential, allowing trees to thrive in soils that would otherwise be too poor to support them. Cherry trees communicate chemically with the world around them. When a leaf is damaged by an insect, the tree releases volatile organic compounds into the air. These compounds serve multiple purposes. Some directly repel the insect or make the leaf less palatable. Others attract predators of the insect, turning the tree into a beacon for help. Neighboring trees can detect these airborne signals and begin producing their own defensive compounds in anticipation of attack. This form of communication is silent and invisible, but it is happening constantly in forests and orchards, a conversation carried on the wind. The wood of cherry trees is prized for its warm color and fine grain. It is used in furniture, musical instruments, and decorative objects. The heartwood, the older inner layers, is reddish-brown and durable, while the sapwood near the bark is paler. As the tree ages, it gradually converts sapwood to heartwood, a process that involves depositing tannins and other compounds that make the wood resistant to decay. This is why older cherry wood is more valuable. It has been transformed by time, just as the tree itself transforms each spring. Cherry bark has horizontal lines called linicelles, small pores that allow gas exchange between the inner tissues and the atmosphere. Even though bark is protective, the tree still needs to breathe. Linicelles are particularly visible on cherry trees, giving the bark a distinctive, striped appearance. In some species, the bark peels in thin horizontal strips, adding texture and visual interest. This peeling is not a sign of distress. It is a natural part of the tree's growth. As the trunk expands, the outer bark cracks and sheds, replaced by new layers from within. Cherry trees can live for many decades, some species reaching 100 years or more. In Japan, there are ancient cherry trees that have been celebrated for centuries, their age and size making them cultural landmarks. One such tree, the Jindai sakura in Yamanashi Prefecture, is estimated to be nearly 2,000 years old. Its trunk is massive and gnarled, supported by wooden props, yet it still blooms each spring. People visit it as they would a temple, standing quietly in its presence, witnessing a living connection to the distant past. These ancient trees remind us that renewal is not just for the young. Even the old can flower. The genome of the cherry tree has been sequenced, revealing the genetic instructions for everything from flower color to disease resistance. Scientists have identified thousands of genes, each playing a role in the tree's development and response to the environment. Some genes are active only during flowering, others only in roots, and still others switch on and off in response to temperature, light, or stress. This genetic map allows researchers to understand how cherry trees adapt and evolve, and it opens possibilities for breeding varieties that are more resilient to climate change, pests, and diseases. It is a kind of blueprint, written in the language of DNA, for renewal itself. Cherry blossoms have inspired scientific studies on the nature of beauty and human emotional response. Researchers have found that viewing cherry blossoms can reduce stress, lower blood pressure, and improve mood. The effect is partly psychological, tied to cultural associations and the joy of spring, but there may also be physiological components. Natural scenes with soft colors and organic shapes engage the brain differently than urban environments, activating areas associated with relaxation and positive emotion. The Japanese practice of hanami, or flower viewing, is not just tradition. It is a form of nature therapy, a deliberate pause to absorb beauty and let it work its quiet influence. The petals of cherry blossoms contain trace amounts of coumarin, the same compound that gives freshly cut hay and vanilla its sweet scent. When petals are dried or preserved in salt, as they are for certain traditional foods, the coumarin becomes more concentrated and noticeable. In Japan, salted cherry blossoms are used to flavor tea, sweets, and rice dishes, adding a subtle floral note. The practice of eating flowers is ancient, found in many cultures, and it reflects a desire to take the beauty of the bloom inward, to make it part of oneself, even if only for a moment. Cherry trees are planted along rivers and in parks, not only for their beauty, but for their role in ecosystems. Their flowers provide early season nectar when few other sources are available, supporting pollinators emerging from winter dormancy. Their leaves feed caterpillars, which in turn feed birds. Their branches offer nesting sites, and their roots stabilize soil along waterways, reducing erosion. A single cherry tree is a hub of life, supporting dozens of species directly and hundreds indirectly. When we plant a cherry tree, we are not just adding decoration. We are weaving a new thread into the ecological fabric. In traditional Chinese medicine, cherry blossoms and other parts of the tree have been used for centuries. The bark was believed to have properties that could soothe coughs and reduce inflammation. Modern research has identified compounds in cherry bark with mild anti-inflammatory and antimicrobial effects, though these are not strong enough to replace conventional medicine. What is interesting is the long history of observation that led people to use these plants. They watched, experimented, and pass down knowledge, building a pharmacopoeia based on careful attention to the natural world. The phenomenon of cherry blossoms blooming earlier in recent decades is one of the most visible signs of climate change. In Kyoto, records show that peak bloom now occurs about a week earlier than it did a century ago. This shift is driven by warmer spring temperatures, which trigger blooming before the calendar suggests it should happen. While earlier blooms might seem harmless, they can create mismatches in ecosystems. If flowers open before pollinators are active, or if late frosts damage the blooms, the tree's reproductive success declines. These changes ripple outward, affecting the species that depend on the tree and altering the timing of ecological events that have been synchronized for millennia. Cherry trees are also sensitive to changes in winter. As winters warm, some regions may no longer provide the chilling hours required for proper bud development. This could push cherry cultivation northward or to higher elevations, leaving behind areas where these trees have been grown for generations. Scientists are working to develop varieties that require fewer chilling hours, adapting the tree to a changing climate. It is a race between human ingenuity and the pace of environmental change, and the outcome is uncertain. What is certain is that the cherry blossoms of the future may not bloom in the same places or at the same times as they do now. Pollination in cherry blossoms is a dance of precision and chance. The flowers produce nectar at the base of their petals, drawing bees into close contact with the stamens and pistil. As a bee moves through the flower, pollen grains stick to its body, carried to the next bloom. Each pollen grain must land on a receptive stigma, germinate, and send a tube down through the style to reach the ovule. Only then can fertilization occur. The process is delicate, and many factors can disrupt it. Cold weather, lack of pollinators, rain that washes pollen away. Yet enough succeed to ensure the next generation. Cherry trees have been hybridized extensively, resulting in hundreds of cultivated varieties. Some have double flowers, with extra petals that create a fuller, more dramatic appearance. Others have weeping branches that cascade like waterfalls. Still others bloom in autumn as well as spring, or produce flowers in shades of yellow and green. Each variety is the result of careful selection, sometimes over many generations, choosing trees with desirable traits and propagating them. This human intervention has expanded the diversity of cherry blossoms far beyond what exists in the wild, creating a living gallery of form and color. The root system of a cherry tree is shallow compared to some other trees, which makes them vulnerable to drought and wind. Most of the roots are in the top meter of soil, where water and nutrients are most available, but also where conditions can change rapidly. During dry periods, cherry trees may drop leaves early or reduce growth to conserve resources. In urban environments, where soil is often compacted and poor, cherry trees face additional stress. Arborists have learned to improve planting conditions by loosening soil, adding organic matter, and ensuring adequate water, giving these trees a better chance to thrive in challenging settings. Cherry blossoms have been the subject of poetry for over 1,000 years, and this literary tradition has influenced how people see and experience the flowers. In Japanese waka and haiku, cherry blossoms often symbolize the fleeting nature of life, beauty that cannot be held. This theme, called mono no aware, is a gentle sadness at impermanence, an awareness that everything passes. The poems do not mourn this fact. They celebrate it, finding depth and meaning in transience. This perspective has shaped cultural attitudes toward cherry blossoms, making the brief bloom not a tragedy, but a profound reminder to pay attention, to be present, to appreciate what is here now. The structure of a cherry blossom is a study in symmetry and efficiency. 5 petals radiate from the center, each evenly spaced, creating a balanced form. This 5-fold symmetry is common in the rose family, to which cherries belong, and it reflects underlying genetic and developmental patterns. The petals are arranged to maximize visibility to pollinators while protecting the reproductive organs at the center. The stamens, usually numerous, surround a single pistil, and their placement ensures that any visiting insect is likely to contact both, facilitating cross-pollination. It is a design refined by millions of years of evolution, simple in appearance but complex in function. Cherry trees respond to light not just in terms of photosynthesis, but also in their growth patterns. They exhibit phototropism, growing toward light sources, and photoperiodism, responding to the length of day and night. These responses are mediated by plant hormones like auxin and phytochrome, which detect light quality and duration. In spring, lengthening days signal the tree to allocate resources toward growth and reproduction. In autumn, shortening days trigger the preparation for dormancy. The tree is constantly reading its environment, adjusting its behavior to align with the seasons. The falling of cherry blossom petals is not passive. The tree actively sheds them by forming an abscission layer at the base of each petal, similar to the process used for leaves. This allows the tree to control when the petals fall, typically after pollination has occurred or when the flower is no longer viable. Wind and rain accelerate the process, but the tree is already prepared for it. The result is that petals fall cleanly, without tearing, creating the graceful drift that so captivates observers. It is a controlled ending, as deliberate as the bloom itself. Cherry blossoms have been introduced to many parts of the world, often as gifts between nations. The cherry trees in Washington, D.C. were a gift from Japan in 1912, a gesture of friendship that has grown into an annual festival, attracting hundreds of thousands of visitors. Similar plantings exist in cities across Europe, Asia, and beyond. These trees carry with them not just beauty, but also stories of connection, diplomacy, and shared appreciation. They are living symbols of relationships between cultures, rooted in soil far from their origin, but thriving nonetheless. The cellular structure of cherry blossom petals is what gives them their texture and translucence. Each petal is composed of layers of epidermal cells on the top and bottom, with mesophyll cells in between. These cells contain vacuoles filled with sap, which can include pigments, sugars, and other compounds. The arrangement and shape of these cells affect how light passes through the petal, creating effects of color and luminosity. When backlit by the sun, cherry blossoms seem to glow, the light diffusing through the thin tissue and illuminating the intricate vein patterns that deliver water and nutrients. Cherry trees have a natural lifespan, and as they age, their growth slows and their vulnerability to disease increases. Older trees often develop hollow trunks, the heartwood decaying while the outer sapwood continues to transport water and nutrients. These hollows can become habitats for birds, insects, and small mammals, adding another layer to the tree's ecological role. In some cases, ancient cherry trees are carefully maintained, with supports added to weakened branches and treatments applied to protect against pests. These efforts reflect a recognition that some trees are more than individuals. They are monuments, witnesses to history, deserving of care. The timing of cherry blossom festivals is a cultural negotiation between nature and human schedules. Organizers must predict peak bloom weeks or months in advance, booking venues, arranging performances, and coordinating logistics. Sometimes the blossoms cooperate, opening right on schedule. Other times, they bloom early or late, and the festival proceeds regardless, a celebration of the tree itself rather than the flowers. This flexibility reflects a deeper understanding. The Cherry Blossom Festival is not just about seeing flowers. It is about gathering, about marking the arrival of spring, about renewing social bonds. The flowers are the occasion, but the people are the heart of it. Cherry trees can be propagated not only by grafting, but also by cuttings and tissue culture. Cuttings involve taking a small branch, treating it with rooting hormone, and encouraging it to develop roots in soil or water. Tissue culture is more advanced, using tiny pieces of plant tissue grown in sterile conditions on nutrient media. From a single bud, thousands of identical plants can be produced. These techniques allow rare or valuable varieties to be preserved and distributed widely, ensuring that genetic diversity and unique traits are not lost. It is a form of conservation, multiplying beauty and resilience through careful science. The scent of cherry blossoms, though subtle, is chemically complex. Researchers have identified dozens of volatile compounds in the fragrance, including alcohols, esters, and terpenes. The exact blend varies by species and even by individual tree, influenced by genetics and environment. Some compounds are sweet and floral, others green and fresh, and together they create a scent that is difficult to describe but easy to recognize. This complexity is part of what makes natural fragrances so appealing and so hard to replicate synthetically. A cherry blossom scent is not a single note, but a chord, a harmony of molecules released into the air. Cherry trees are sometimes planted in memory of loved ones, becoming living memorials. These trees grow and change over the years, their annual blooms a recurring reminder of the person honored. There is something deeply comforting in this practice, the idea that life continues, that beauty returns, that renewal is built into the fabric of the world. The tree does not replace what was lost, but it offers a place to return to, a focus for remembrance and reflection. Each spring, the flowers come back, and with them, the memories. The energy required to produce a cherry blossom is significant. The tree must allocate stored carbohydrates from the previous year, along with water and minerals absorbed by the roots. Each flower represents an investment, a gamble that conditions will allow for pollination and seed production. If the flowers are damaged by frost or fail to attract pollinators, that energy is lost. Yet the tree makes this investment every year, driven by evolutionary imperatives that have proven successful over millennia. It is a reminder that renewal requires resources, effort, and risk. Nothing blooms without cost. Cherry blossoms have been studied for their effects on urban microclimates. Trees in cities provide shade, reduce temperatures through evapotranspiration, and filter air pollutants. During bloom, cherry trees contribute to these benefits while also enhancing the aesthetic and psychological environment. Studies have shown that streets lined with flowering trees are perceived as more pleasant and are associated with higher property values and increased foot traffic. The presence of cherry blossoms can transform a utilitarian space into a destination, drawing people outdoors and fostering a sense of community. The tree's influence extends beyond biology into the social and economic fabric of urban life. The relationship between cherry trees and their pollinators is not guaranteed. In areas where native bee populations have declined due to habitat loss, pesticides, or disease, cherry trees may struggle to produce fruit. This has led to increased interest in supporting pollinator health through planting wildflowers, reducing chemical use, and creating habitat corridors. The health of cherry trees and the health of bees are intertwined, each dependent on the other. It is a partnership that has existed for millions of years, and its disruption is a warning sign that ecosystems are under strain. Cherry trees grown for fruit, such as sweet cherries and sour cherries, are closely related to ornamental varieties but have been selected for different traits. Fruit cherries are bred for larger, fleshier drupes with higher sugar content, while ornamental cherries are bred for flower size, color, and abundance. Some ornamental varieties produce small, bitter fruits that are not palatable to humans but are eaten by birds. This distinction between fruit and flower is a human creation, a divergence of purpose imposed through selective breeding. In the wild, The tree makes no such distinction. The flower's purpose is always the fruit, and the fruit's purpose is always the seed. The bark of cherry trees contains compounds that deter some herbivores, but not all. Certain caterpillars have evolved to tolerate or even require these compounds, feeding exclusively on cherry leaves. This specialization is both a strength and a vulnerability. The caterpillar has access to a food source that others cannot use, but is also entirely dependent on the presence of cherry trees. If the trees disappear, so does the caterpillar. These tight ecological relationships are common in nature, and they highlight the interconnectedness of species. The loss of one can cascade through the web, affecting many others. Cherry blossoms have been depicted in art for centuries, from ancient Chinese scroll paintings to Japanese woodblock prints to contemporary photography. Each medium captures different aspects of the bloom. The delicate line work of ink paintings emphasizes grace and simplicity. Woodblock prints often show blossoms in bold, flat colors against dramatic skies, and photography can freeze a single petal mid-fall or capture the play of light through translucent petals. These artistic representations shape how we see the actual flowers, creating a feedback loop between nature and culture. We see the blossoms through the lens of art, and art is inspired by the blossoms, each enriching the other. The wind that moves through cherry branches carries more than just fragrance and pollen. It carries information encoded in pressure waves and chemical signals, information that other trees can detect and respond to. When cherry blossoms release their volatile compounds into the air, neighboring plants sometimes adjust their own flowering schedules, their own defensive chemistry, as if listening to a conversation carried on currents too subtle for human perception. The forest, the orchard, the planted avenue of ornamental cherries all exist within webs of communication we are only beginning to understand. Scientists studying plant communication have discovered that trees can detect ethylene gas released by their neighbors, a signal that often indicates stress or ripening or the approach of autumn. Cherry trees produce ethylene as their flowers age, as petals prepare to fall, and this molecular whisper may influence the timing of nearby plants. The air around a blooming cherry grove becomes a complex mixture of signals, a chemical dialogue written in molecules that drift and disperse and bind to receptors on leaves and stems throughout the surrounding landscape. The way cherry petals fall has been studied not just by poets, but by physicists interested in fluid dynamics and the mathematics of descent. A falling cherry petal does not drop straight down like a stone. It tumbles and spirals, catching air in ways that slow its fall and carry it laterally, sometimes dozens of meters from the parent tree. This tumbling motion follows patterns that can be described mathematically, patterns similar to those seen in falling leaves, in certain seeds, in the descent of snowflakes through still air. Researchers using high-speed cameras have captured the exact motion of falling petals, analyzing how their shape and flexibility create vortices in the air, tiny whirlwinds that both slow the fall and impart spin. The petal acts as a kind of wing, generating lift even as it descends, and this lift allows it to travel farther from the tree, increasing the area over which fallen petals will eventually decompose and return nutrients to the soil. What appears as simple beauty serves a function, spreading organic matter across a wider territory, enriching the ground in patterns determined by wind and physics. The decomposition of cherry petals happens quickly. Within days of falling, the thin tissue begins to break down, colonized by bacteria and fungi that consume the simple sugars and starches still present in the cells. This rapid decomposition is partly due to the thinness of the petals. There is little structural material to resist decay, and partly due to the chemistry of the tissue itself, which contains relatively few defensive compounds compared to leaves or bark. As petals decompose, they release nitrogen back into the soil, nitrogen that was invested in the proteins and enzymes of the flower cells. This nitrogen becomes available to the tree's roots, to the roots of nearby plants, to the microorganisms that populate the soil ecosystem. The tree, in a sense, recycles part of its investment, though much of the energy and material in the flowers is lost to the wind, to pollinators, to the broader environment. This is the cost of sexual reproduction, the price paid for genetic mixing and the possibility of adaptation. Cherry trees also reproduce clonally, through root suckers that emerge from the lateral roots, spreading underground. These suckers are genetically identical to the parent tree, clones that can grow into full-size trees if conditions allow. In wild cherry populations, you often find clusters of trees that are actually a single genetic individual, a colony connected underground, sharing resources through root grafts and mycorrhizal networks. This clonal growth provides a form of insurance, a way for the tree's genes to persist even if the main trunk is damaged or destroyed. The balance between sexual and asexual reproduction varies among cherry species and depends on environmental conditions. In stable, favorable environments, clonal growth may dominate, producing dense thickets of genetically identical stems. In disturbed or changing environments, sexual reproduction becomes more important, generating genetic diversity that increases the likelihood that some offspring will be adapted to new conditions. The tree, without consciousness or intent, employs both strategies, responding to signals in the soil and air that indicate which approach is more likely to succeed. Cherry wood has been valued by humans for thousands of years, prized for its fine grain, its warm reddish color, and its workability. The wood is moderately hard, dense enough to take a smooth finish, but not so hard that it dulls tools quickly. It has been used for furniture, for musical instruments, for decorative carving, for the handles of tools and utensils. The same tree that produces ephemeral flowers produces wood that can last for centuries if properly cared for, a different kind of persistence. The color of cherry wood deepens with age and exposure to light, a process called photochemical aging. Ultraviolet light causes changes in the lignin and other compounds in the wood, shifting the color from a pale pinkish tan to a deeper reddish brown over years and decades. This aging is considered desirable in woodworking, a sign of authenticity and quality, and furniture makers sometimes expose cherry wood to controlled amounts of sunlight to accelerate the process. Inside the wood, the growth rings tell the story of the tree's life, each ring representing 1 year of growth, one cycle of spring and summer and autumn and winter. Wide rings indicate years of abundant water and warmth, years when the tree grew vigorously. Narrow rings indicate years of stress, of drought or cold or competition. By reading these rings, dendrochronologists can reconstruct past climates, can date wooden objects, can trace the history of forests and the impact of human activity on tree growth. Cherry trees can live for many decades, though their lifespan varies by species and growing conditions. Wild cherries in forests may live for 60 to 80 years, sometimes longer if conditions are favorable and competition is not too intense. Ornamental cherries, especially grafted varieties, often have shorter lifespans, 30 to 40 years being typical, though individual trees sometimes persist much longer. The famous cherry trees in Washington, D.C., many of which were planted in 1912, are now well over a century old, ancient by the standards of ornamental cherries, and carefully managed to extend their lives as long as possible. As cherry trees age, their flowering often becomes less profuse. The number of flower buds produced each year may decline, and the tree's energy increasingly goes into maintaining existing structure rather than producing new growth. This is a natural part of the aging process in trees, a gradual shift in resource allocation that reflects the changing balance between growth and maintenance, between reproduction and survival. Old cherry trees develop character that young trees lack. Their bark becomes deeply furrowed, textured with the accumulated scars of decades. Their branches thicken and twist, shaped by years of wind and snow and the weight of their own growth. Cavities may form where branches have broken off, providing habitat for nesting birds, for roosting bats, for the insects and fungi that colonize deadwood. An old cherry tree becomes an ecosystem in itself, supporting life in ways that a young tree cannot. The phenomenon of cherry blossom viewing, hanami, has influenced not just culture, but also urban planning and landscape design in many countries. Cities plant cherry trees along streets and in parks specifically to create spaces for public enjoyment during bloom season. These plantings are often carefully planned to include multiple varieties that bloom at different times, extending the period of flowering and reducing the risk that a single storm will destroy the entire display. In Japan, the cherry blossom forecast, sakura zensun, is a serious matter, reported in national news and followed closely by millions of people planning trips and gatherings. Meteorological agencies use historical data, current weather patterns, and observations from volunteer networks to predict bloom dates for hundreds of locations across the country. The forecasts are updated daily as the bloom front moves northward with a warming spring, a wave of pink and white that sweeps across the archipelago over the course of several weeks. This mass coordination around a natural event creates a shared cultural experience, a collective participation in the rhythms of the season. It also generates economic activity, travel, hospitality, food and beverage sales, photography equipment, special products marketed for the brief bloom period. The cherry blossom season has become a significant economic phenomenon, worth billions of dollars annually in Japan alone, a remarkable example of how a natural event can shape human society and economy. The practice of hanami dates back over 1,000 years, originally associated with the imperial court and aristocracy, later spreading to samurai, and eventually to all levels of society. Early hanami focused on plum blossoms, which bloom earlier than cherries, but by the Heian period, cherry blossoms had become the dominant focus. The practice involves gathering beneath blooming trees to appreciate their beauty, to eat and drink, to compose poetry, to enjoy the company of friends and family. Traditional hanami includes elements of ritual and etiquette, the spreading of blankets or tarps beneath the trees, the sharing of special foods, the consumption of sake, the composition of poems. These practices create a framework for attention, a way of marking the event as special, as worthy of mindfulness. In modern times, hanami can range from quiet contemplation to exuberant parties, but the core remains the same: a deliberate pause to witness beauty, to acknowledge the season, to participate in a tradition that connects the present to centuries of past observers. The foods associated with hanami often incorporate cherry blossoms or cherry leaves, or a colored pink to evoke the flowers. Sakura machi, a sweet rice cake wrapped in a pickled cherry leaf, is a traditional hanami food. The leaf is edible, slightly salty, providing a contrast to the sweet rice and bean paste filling. Dango, skewered rice dumplings, are often colored pink, white, and green to represent cherry blossoms, snow, and new leaves. These foods are seasonal, available only during bloom time, reinforcing the sense of the cherry blossom period as a distinct and special moment in the year. Cherry blossoms have been depicted in art for centuries, appearing in paintings, prints, textiles, ceramics, and countless other media. In Japanese art, cherry blossoms are one of the most common motifs, symbolizing beauty, transience, and the arrival of spring. Artists have developed conventions for depicting the flowers, the number of petals, the arrangement of branches, the inclusion of birds or other elements that allow viewers to recognize cherry blossoms even in highly stylized representations. The famous woodblock prints of the Edo period often feature cherry blossoms, sometimes as the main subject, sometimes as background elements that establish season and mood. Artists like Hiroshige and Hokusai created numerous prints showing famous cherry blossom viewing sites, capturing not just the trees but the crowds of people gathered beneath them, the atmosphere of celebration and appreciation. These prints were popular souvenirs and decorative objects, spreading images of cherry blossoms throughout Japan and eventually to the rest of the world. In Western art, cherry blossoms became a subject of interest in the late 19th and early 20th centuries, as Japanese art and aesthetics influenced European and American artists. The Impressionists and Post-Impressionists were particularly drawn to the delicate colors and the challenge of capturing the ephemeral quality of the blooms. Cherry blossoms appeared in paintings, in decorative arts, in garden design, as part of a broader fascination with Japanese culture that influenced Western aesthetics in lasting ways. The symbolic meaning of cherry blossoms has varied across cultures and time periods. In Japan, the flowers are associated with mono no aware, a term that describes a sensitivity to the transience of things, a gentle sadness at the passing of beauty. This concept is central to Japanese aesthetics and philosophy, informing not just attitudes toward cherry blossoms, but toward life itself. The flowers, beautiful and brief, become a meditation on impermanence, a reminder that all things change and pass away. In other contexts, cherry blossoms have symbolized renewal, hope, the arrival of spring, the beauty of youth. During the samurai era, cherry blossoms were sometimes associated with the warrior's readiness to die, the idea that life should be lived fully and beautifully, ending at the peak of vitality rather than declining into old age. This association was later appropriated and distorted by militaristic ideologies, a reminder that symbols can be used in many ways, their meanings shaped by context and intent. Today, cherry blossoms are widely recognized as symbols of peace, friendship, and international goodwill, partly due to the gift of cherry trees from Japan to the United States in 1912 and similar gifts to other countries. The trees planted in Washington, D.C. have become one of the city's most beloved features, attracting millions of visitors each spring and serving as a living symbol of the relationship between the two nations. Similar plantings in other cities around the world serve comparable symbolic functions: trees as diplomats, beauty as a bridge between cultures. The chemistry of cherry blossom fragrance is complex, involving dozens of volatile compounds released by the flowers. The scent is generally described as delicate, sweet, slightly almond-like, though it varies among species and even among individual trees. Not all cherry blossoms are fragrant. Some ornamental varieties have little or no scent, but those that do produce fragrance release a mixture of compounds that includes benzaldehyde, linalool, and various esters and alcohols. Benzaldehyde is the compound primarily responsible for the almond-like note in cherry blossom fragrance. It is also found in almonds, in apricot kernels, in the leaves and bark of cherry trees. This compound is produced through the breakdown of amygdalin, a cyanogenic glycoside that serves as a defensive compound in many plants in the Prunus genus. In small amounts, as volatilized from flowers, benzaldehyde is pleasant and harmless. In larger concentrations, as found in crushed seeds, the related compounds can release cyanide and are toxic. Linalool is a terpene alcohol with a floral, slightly spicy scent, found in many flowers, and also in lavender, coriander, and numerous other plants. It contributes to the sweet, floral character of cherry blossom fragrance and is a common component in perfumes and scented products. Linalool has been studied for its effects on the nervous system and has been found to have mild sedative and anxiolytic properties in animal studies, suggesting that the scent of flowers may have subtle physiological effects on those who smell them. The fragrance of cherry blossoms is most intense during warm, sunny days when the flowers are actively attracting pollinators. The volatile compounds evaporate more readily at higher temperatures, creating a stronger scent that can carry on the breeze. In cool or cloudy conditions, the fragrance is more subtle, the chemical signals less urgent. This temperature-dependent release of scent is common among flowers and represents another way that plants respond to environmental conditions, adjusting their strategies to match the likelihood of pollinator activity. Human perception of cherry blossom fragrance varies widely. Some people find the scent enchanting, evoking memories of spring and renewal. Others find it faint or unremarkable, barely noticeable among the many scents of the season. This variation reflects differences in individual olfactory sensitivity, in past experiences and associations, in a specific compounds present in a given tree's flowers. Scent is deeply personal, tied to memory and emotion in ways that visual beauty is not, and the same fragrance can evoke entirely different responses in different people. The petals of cherry blossoms contain anthocyanins, the same pigments found in many fruits and flowers, responsible for red, pink, and purple colors. The specific shade of pink varies depending on the concentration and type of anthocyanins, on the pH of the cell sap, and on the presence of other pigments and copigments that modify color. White cherry blossoms lack anthocyanins, their petals reflecting light without selective absorption in the visible spectrum, appearing white because they scatter all wavelengths equally. Anthocyanins serve multiple functions in flowers. They attract pollinators, particularly insects that can see in the ultraviolet range and perceive patterns invisible to human eyes. They protect the delicate reproductive structures from UV damage, absorbing harmful wavelengths before they can damage DNA and proteins. And they may have antioxidant properties, protecting cells from oxidative stress during the metabolically intense period of flowering. The production of anthocyanins is regulated by genes that respond to light, temperature, and developmental signals. In cherry trees, the intensity of petal color can vary depending on growing conditions, with cooler temperatures during bud development sometimes producing deeper colors. This is why cherry blossoms in cooler climates or higher elevations sometimes appear more intensely colored than those in warmer regions, a subtle variation that adds to the diversity of the bloom experience. Cherry blossoms are edible, though they are not particularly nutritious, and are more often used for their aesthetic and symbolic value than for sustenance. In Japan, cherry blossoms are sometimes preserved in salt and used as a garnish or ingredient in special dishes. The salted blossoms, called sakura no shiizuke, are rinsed before use and added to rice, tea, or sweets, imparting a subtle floral flavor and a visual reminder of spring. Sakura tea, made by steeping salted cherry blossoms in hot water, is served at weddings and other celebrations. The blossoms unfurl in the hot water, floating in the cup like tiny flowers in a pond, creating a beautiful and symbolic beverage. The tea itself is mildly salty and floral, more about the experience and the symbolism than about flavor. Drinking sakura tea is a way of literally consuming the season, of taking the beauty of spring into the body, a poetic gesture that connects the physical and the symbolic. Cherry leaves are also used in Japanese cuisine, particularly for wrapping sakura mochi. The leaves are pickled in salt, which preserves them and develops their flavor. The pickling process breaks down some of the compounds in the leaves, including coumarin, which gives the pickled leaf a sweet, vanilla-like aroma. This scent is distinctive and immediately recognizable to anyone familiar with sakura mochi, a smell that evokes spring and tradition. The use of cherry blossoms and leaves in food is an example of shun, the Japanese concept of seasonality in cuisine. Shun emphasizes eating foods at their peak, when they are freshest and most flavorful, and it extends beyond ingredients to include seasonal presentations and preparations. Cherry blossoms appear in food only during their brief season, reinforcing the connection between eating and the cycles of nature between nourishment and time. Cherry trees support a wide variety of wildlife beyond the pollinators that visit their flowers. Birds nest in their branches, feed on their fruits, and glean insects from their bark and leaves. Mammals, from squirrels to deer to bears, eat the cherries when they ripen, dispersing seeds in their droppings. Insects of many kinds feed on various parts of the tree: leaves, flowers, sap, wood. And these insects in turn feed birds, spiders, and other predators. The cherries produced by wild cherry trees are generally small and tart, with a large pit relative to the amount of flesh. These fruits are not the sweet cherries cultivated for human consumption, but they are nutritious for wildlife, high in sugars and containing vitamins and minerals. Birds are the primary dispersers of wild cherry seeds, eating the fruits whole and later excreting the seeds at a distance from the parent tree. The seeds pass through the bird's digestive system unharmed, sometimes even benefiting from the scarification that aids germination. Some cherry species produce fruits that contain higher levels of cyanogenic compounds, making them bitter or even mildly toxic to some animals. These compounds deter some potential seed predators while allowing others, particularly birds, to consume the fruits safely. Birds generally have higher tolerance for these compounds than mammals do, and they process the fruits quickly, reducing exposure time. This selective toxicity helps ensure that seeds are dispersed by the most effective vectors, by animals that will carry them far and deposit them in conditions favorable for germination. The timing of cherry fruit ripening is coordinated with the life cycles of the birds that disperse them. In many regions, cherries ripen in early to mid-summer, when migratory birds are raising young and need abundant food. The trees, in a sense, offer a resource at the time when it will be most eagerly consumed and most effectively dispersed. This synchronization is the result of evolutionary processes that have shaped both the trees and the birds, creating a mutualism that benefits both. Cherry trees are susceptible to various diseases and pests, challenges that can affect their health, their flowering, and their lifespan. Fungal diseases like black knot, brown rot, and powdery mildew can damage branches, fruits, and leaves. Bacterial diseases like bacterial canker can cause dieback and weeping sores on the bark. Insect pests like aphids, borers, and caterpillars can defoliate trees, damage wood, or spread diseases. Despite these challenges, cherry trees have persisted for millions of years, adapting to pests and diseases through natural selection, developing defenses and tolerances that allow them to survive and reproduce. Wild cherry populations contain genetic diversity that includes resistance to various threats, a reservoir of adaptive potential that cultivated varieties often lack. This is why wild populations are important for conservation, not just for their own sake, but as sources of genetic material that may be needed to breed resilient trees for the future. Climate change poses new challenges for cherry trees, altering the temperature and precipitation patterns that govern their life cycles. Warmer winters may not provide sufficient chilling hours, leading to poor flowering or erratic bloom timing. Earlier springs may cause trees to bloom before pollinators are active, or expose flowers to late frosts that damage the delicate tissue. Shifts in the timing of seasons can disrupt the synchronization between trees and the animals that depend on them, unraveling ecological relationships that have existed for millennia. Researchers are studying how cherry trees are responding to climate change, monitoring bloom dates, tracking range shifts, and investigating the genetic basis of adaptability. Some cherry populations are already showing signs of adaptation, with trees that bloom earlier or that require fewer chilling hours becoming more common. This is evolution in action, natural selection favoring traits that match the changing environment. But evolution takes time, and the pace of climate change may exceed the capacity of some populations to adapt, particularly in fragmented landscapes where gene flow is limited. Conservation efforts for cherry trees include protecting wild populations, preserving genetic diversity, and establishing seed banks and living collections. Botanical gardens and arboreta maintain collections of cherry species and varieties, ensuring that genetic material is available for research and for future restoration efforts. These collections are not just repositories of beauty, but libraries of adaptive potential, resources that may be crucial as we navigate an uncertain future. The study of cherry blossoms has contributed to broader scientific understanding in fields ranging from genetics to climate science to the ecology of mutualisms. Research on the genetic control of flowering time in cherries has provided insights applicable to many other plants, including crops. Studies of cherry blossom phenology have helped refine climate models and improve our understanding of how ecosystems respond to environmental change. Investigations of pollination in cherry trees have revealed complexities in plant-pollinator interactions that inform conservation strategies for both plants and pollinators. Cherry blossoms also serve as a subject for citizen science, with volunteers around the world recording bloom dates, photographing trees, and contributing observations to databases that scientists use to track phenological changes. These contributions are valuable because they provide data from many locations over many years, creating records that would be impossible for professional scientists to gather alone. Citizen science projects around cherry blossoms engage people with nature, with science, and with the seasons, fostering awareness and stewardship. The act of observing cherry blossoms, of noting when they bloom and how long they last, is a form of attention that connects us to place and to time. It requires us to notice, to remember, to compare this year with last year, to recognize patterns and changes. This kind of attention is increasingly rare in modern life, where many people spend most of their time indoors, disconnected from the rhythms of the natural world. Cherry blossoms, because they are so beautiful and so brief, invite us back into that rhythm, even if only for a few days each spring. The cellular architecture of cherry blossom petals reveals itself under magnification as a landscape of extraordinary delicacy. Each petal consists of two epidermal layers sandwiching a middle layer of mesophyll cells, creating a structure typically only 20 to 30 cells thick. These cells are arranged with spaces between them, tiny air pockets that scatter light and contribute to the soft, almost luminous quality of the petals. The upper epidermis often has a slightly different texture than the lower, with the upper surface sometimes bearing microscopic papillae, tiny dome-shaped protrusions that enhance the petal's ability to reflect certain wavelengths of light. This microscopic topography influences not just color, but also how the petal feels to touch, creating that characteristic silk-like texture. The thinness of the petal allows light to pass partially through, so that when you hold a cherry blossom up to the sky, you see not just reflected color, but also transmitted light, a gentle glow that seems to come from within the flower itself. Within these delicate cells, the machinery of life continues even as the flower blooms. Mitochondria generate energy to maintain cellular processes, though at a gradually declining rate as the flower ages. Vacuoles, large fluid-filled compartments within each cell, maintain turgor pressure that keeps the petal firm and extended. When these vacuoles begin to lose water, the petal starts to wilt, one of the first visible signs of senescence. The cell walls are composed primarily of cellulose, hemicellulose, and pectin, creating a flexible yet structured framework. Unlike woody tissues, petal cell walls remain relatively thin and pliable, allowing the petal to bend and flutter in the slightest breeze without tearing. This flexibility is part of what makes cherry blossoms so responsive to wind, creating that characteristic gentle movement that has captivated observers for centuries. The pigments within petal cells are not uniformly distributed, but concentrated in specific cellular compartments. Anthocyanins reside in the vacuoles, where the acidic pH helps stabilize their color. The concentration of these pigments can vary not just between different cultivars, but also within a single petal, creating subtle gradations of color from the base to the tip, or from the center outward. Some cherry varieties show a phenomenon called color intensification, where petals deepen in hue as they age, the result of continued anthocyanin synthesis even after the flower opens. Others fade as cellular processes slow and pigments gradually break down. These changes happen slowly enough that you might not notice them from one day to the next, but photographs taken across a week of blooming reveal the transformation, a visual record of the flower's brief life. The surface of a cherry blossom petal, when viewed at high magnification, reveals a complex microstructure. The epidermal cells are not flat but slightly convex, creating a gentle undulating surface. This topography affects how light interacts with the petal, contributing to its characteristic sheen. Some cells contain specialized structures called plastoglobuli, small lipid droplets within the chloroplasts and chromoplasts that can affect color perception. The cuticle, a waxy layer covering the epidermis, is remarkably thin on cherry petals compared to leaves, typically only a few micrometers thick. This thinness contributes to the petal's delicate nature, but also makes it more vulnerable to water loss and physical damage. The cuticle's composition includes various waxes, cutin, and sometimes small amounts of aromatic compounds that contribute to the flower's fragrance. Beneath the visible surface, the vascular system of the petal forms an intricate network. Tiny veins branch from the petal base, carrying water and nutrients to all parts of the petal tissue. These veins are visible in some cherry varieties as delicate lines, slightly darker or lighter than the surrounding tissue. The vascular bundles contain xylem cells that transport water upward from the stem and phloem cells that distribute sugars and other nutrients. The pattern of these veins is not random, but follows mathematical principles similar to those seen in leafination, optimizing the distribution of resources while minimizing the amount of structural tissue needed. This efficiency is part of what allows the petal to be so thin and delicate while still maintaining its function. The process of petal abscission, the natural detachment of the flower from the tree, involves a specialized layer of cells at the base of the flower called the abscission zone. As the flower ages, hormonal changes trigger the production of enzymes in this zone that begin to break down the cell walls connecting the flower to the stem. Cellulase and polygalactorinase enzymes gradually dissolve the pectin and cellulose that hold the cells together. This process is carefully regulated, proceeding slowly enough that the flower doesn't simply fall off prematurely, but quickly enough that it detaches cleanly once senescence is advanced. The abscission zone also forms a protective layer of cells on the stem side, which will seal over after the flower falls, preventing infection and water loss from the wound site. This protective layer, called the abscission scar, is a tiny mark on the twig where the flower once attached, a subtle reminder of what was there. The timing of abscission is influenced by multiple factors. Successful pollination can accelerate the process, as the tree redirects resources from maintaining the flower to developing the fruit. Unpollinated flowers may persist slightly longer, continuing to produce attractants in hopes of receiving pollen. Environmental stresses, such as high winds, heavy rain, or sudden temperature changes can trigger earlier abscission, as the tree prioritizes survival over reproduction. The hormone ethylene plays a central role in this process, with ethylene production increasing as the flower ages. This same hormone is involved in fruit ripening and leaf senescence, part of a broader system that plants use to regulate the life cycles of their various parts. Cherry trees demonstrate remarkable plasticity in their reproductive strategies, adjusting their investment in flowering based on environmental conditions and resource availability. In years following abundant flowering and heavy fruit set, a tree may produce fewer flowers the next spring, a phenomenon called alternate bearing. This happens because fruit production is metabolically expensive, depleting the tree's stored carbohydrates and nutrients. The tree needs time to rebuild these reserves before investing heavily in reproduction again. Some cherry varieties show stronger alternate bearing patterns than others, with certain cultivars producing reliable annual displays, while others fluctuate more dramatically. Wild cherry species often show more pronounced alternate bearing than cultivated ornamental varieties, which have been selected partly for consistent flowering. The root system of a cherry tree extends far beyond the visible canopy, sometimes reaching 2 to 3 times the diameter of the crown. These roots serve multiple functions beyond simply anchoring the tree and absorbing water. The fine root tips, barely thicker than a hair, are the primary sites of water and nutrient uptake. These roots are covered with even finer root hairs, microscopic extensions of root epidermal cells that dramatically increase the surface area available for absorption. A single cherry tree may have millions of these root hairs, creating an enormous interface with the soil. These structures are ephemeral, constantly dying and being replaced as the root tip grows through the soil, leaving behind older root sections that become more woody and structural. The relationship between cherry roots and soil fungi extends to multiple species of mycorrhizal partners. Different fungi may associate with the same tree, each contributing slightly different benefits. Some are more efficient at accessing phosphorus, others at mobilizing nitrogen or micronutrients. The fungal community associated with a cherry tree's roots changes with soil conditions, season, and tree age. Young trees often have different mycorrhizal partners than mature trees, reflecting changing nutritional needs as the tree develops. This fungal network also connects individual trees, creating what some researchers call a wood wide web, through which trees can share resources and even chemical signals. A mature cherry tree might be connected through fungal networks to dozens of other trees, both of the same species and different species, creating an underground community that mirrors and supports the visible forest above. The bark of a cherry tree is more than just protective covering. It's a complex tissue with multiple layers, each serving specific functions. The outer bark, which we see and touch, consists of dead cells impregnated with suberin, a waxy substance that makes the tissue water-resistant. This outer layer protects the living tissues beneath from physical damage, temperature extremes, and desiccation. The characteristic horizontal lenticels, those striped markings on cherry bark, are actually pores that allow gas exchange between the living tissues inside the tree and the atmosphere. Oxygen needs to reach the living cells of the inner bark and cambium, and carbon dioxide produced by cellular respiration needs to escape. These lenticels expand and contract slightly with humidity changes, opening more in moist conditions and closing somewhat in dry weather. Beneath the outer bark lies the phloem, the living tissue that transports sugars and other organic compounds from the leaves to all other parts of the tree. This tissue is remarkably thin, often just a few millimeters thick, yet it carries all the photosynthetic products that fuel the tree's growth and metabolism. The phloem consists of specialized cells called sieve tube elements, arranged end to end to form continuous tubes through which sugars flow. These cells are alive but lack nuclei, having lost them during development to maximize internal space for transport. Companion cells adjacent to each sieve tube element retain their nuclei and provide metabolic support, managing the function of the sieve tube elements they serve. This partnership between cells, one specialized for transport and one for metabolic management, is an elegant solution to the challenge of moving nutrients through a large organism. The cambium, a thin layer of actively dividing cells between the phloem and xylem, is where the tree's radial growth occurs. Throughout the growing season, cambial cells divide, producing new xylem cells toward the inside and new phloem cells toward the outside. This process adds a new growth ring each year, the familiar pattern visible in cross-sections of tree trunks. In cherry trees growing in temperate climates, these rings show clear seasonal patterns. Wood produced in spring, when water is abundant and growth is rapid, has larger cells with thinner walls, appearing lighter in color. Summer wood, produced under drier conditions with slower growth, has smaller, thick-walled cells, appearing darker. This alternation of light and dark wood creates the visible ring pattern, with each ring representing 1 year of growth. By counting these rings and measuring their width, researchers can determine a tree's age and reconstruct the environmental conditions it experienced throughout its life. Cherry wood has been valued for centuries for its working properties and aesthetic qualities. The heartwood, the older, central wood that no longer transports water, often develops a warm reddish-brown color due to the deposition of various compounds, including tannins and other phenolics. This color deepens with age and exposure to light, a process called patination. The wood has a fine, even texture and a subtle natural luster that becomes more pronounced when the wood is finished. It's moderately hard and strong, easier to work than oak or maple, but more durable than pine or poplar. These properties have made cherry wood popular for furniture, musical instruments, and decorative items. The wood also has a pleasant, subtle fragrance when freshly cut, a characteristic scent that comes from the same aromatic compounds found in the tree's flowers and leaves. The seasonal cycle of a cherry tree involves profound physiological changes that affect every part of the organism. As autumn approaches and day length shortens, the tree begins preparing for winter dormancy. Photosynthesis gradually slows in the leaves as chlorophyll breaks down, revealing other pigments and creating the yellow and orange colors of autumn foliage in some cherry species. The tree withdraws valuable nutrients from the leaves, transporting nitrogen, phosphorus, and other elements back into the woody tissues for storage. This nutrient salvage is remarkably efficient, with the tree recovering up to 70% of the nitrogen and phosphorus from its leaves before they fall. These recovered nutrients are stored in the roots and woody tissues, ready to support the next spring's growth. As temperatures drop, the tree undergoes a process called cold acclimation, during which cells throughout the tree adjust their composition to survive freezing. Water is withdrawn from cells into intercellular spaces, reducing the risk of ice crystals forming inside cells and rupturing their membranes. The concentration of sugars, proteins, and other dissolved substances in the remaining cellular fluid increases, lowering the freezing point much like antifreeze in a car radiator. Cell membranes undergo compositional changes, incorporating more unsaturated fatty acids that remain flexible at lower temperatures. These adaptations allow the tree's tissues to survive temperatures well below freezing without damage. Different parts of the tree have different levels of cold hardiness, with buds and woody tissues generally more cold tolerant than roots, which are somewhat insulated by the soil. The tree's water transport system also undergoes changes to survive winter. In summer, water moves through the xylem in continuous columns, held together by cohesion and adhesion forces in a process called the cohesion tension theory. But freezing temperatures can cause these water columns to break, forming air bubbles called embolisms that block water transport. To prevent catastrophic failure of the water transport system, cherry trees and other temperate woody plants have evolved several strategies. The xylem vessels are relatively narrow, limiting the spread of embolisms. The tree also maintains redundant pathways, so that if some vessels become blocked, water can flow through others. In spring, as temperatures rise and sap begins to flow, the tree can repair some embolisms by dissolving the air bubbles back into solution under positive root pressure. The spring awakening of a cherry tree is triggered by a complex interplay of temperature and photoperiod. As soil temperatures rise, root activity resumes, beginning to absorb water and nutrients. This creates a positive pressure that pushes sap upward through the xylem, a phenomenon you can observe in maple trees as the sap flow that's harvested for maple syrup. Cherry trees show a similar, but less pronounced sap flow in early spring. As this water reaches the buds, it delivers hormones and nutrients that were stored in the roots over winter, signaling that it's time to break dormancy. The bud scales begin to swell as cells inside resume growth, and the metabolic rate of the entire tree increases dramatically. Within the buds, the miniature leaves and flowers that formed the previous summer begin to expand, their cells elongating and dividing rapidly. The energy for this spring growth surge comes partly from stored carbohydrates, primarily starch that was accumulated in the roots, trunk, and branches during the previous growing season. Specialized cells called ray parenchyma, which extend radially through the wood, store these carbohydrates and mobilize them when needed. As temperatures rise, enzymes convert the stored starch back into sugars, which are transported to the growing buds and other active tissues. This mobilization of stored resources is so significant that the sugar concentration in the tree's sap increases noticeably in early spring. The tree is essentially living off its savings, investing stored capital to produce the leaves that will generate new income through photosynthesis. This is why the previous year's growing conditions affect the current year's flowering. A tree that accumulated abundant reserves will have more resources to invest in reproduction. The development of leaves and flowers follows overlapping but distinct timelines in cherry trees. Flower buds typically open first, before the leaves are fully expanded. This timing ensures that the flowers are clearly visible to pollinators and not hidden by foliage. It also means that the tree must support early flower development entirely on stored resources, as the leaves aren't yet photosynthesizing. This is a significant investment, and it explains why environmental stresses during the previous growing season can reduce flowering the following spring. The tree simply doesn't have enough stored reserves to support abundant flower production. Once the flowers fade and leaves expand, the tree enters its primary growth phase, with photosynthesis generating the energy and carbon compounds needed for all other activities. Cherry leaves are remarkably efficient photosynthetic organs, with their structure optimized for capturing light and exchanging gases. The leaf blade is typically thin, allowing light to penetrate to the chloroplast-containing cells inside. The upper surface often has a waxy cuticle that reflects some light, reducing heat absorption and water loss. The lower surface has numerous stomata, microscopic pores that open to allow carbon dioxide to enter for photosynthesis and water vapor to exit as a byproduct of transpiration. A single cherry leaf may have several hundred stomata per square millimeter, each one a tiny valve that the plant can open or close in response to environmental conditions. On hot, dry days, the stomata partially close to conserve water, even though this reduces the rate of photosynthesis. On cool, humid days, they open wider, maximizing carbon dioxide uptake and photosynthetic rate. The internal structure of a cherry leaf reveals elegant organization. Just below the upper epidermis lies a layer of palisade mesophyll cells, elongated cells packed with chloroplasts and arranged perpendicular to the leaf surface. This arrangement maximizes light capture, with each cell positioned to intercept incoming photons. Below the palisade layer is the spongy mesophyll, a more loosely arranged tissue with air spaces between the cells. These air spaces allow carbon dioxide to diffuse from the stomata to the photosynthesizing cells and allow oxygen, a byproduct of photosynthesis, to diffuse out. The air spaces also make the leaf interior somewhat compressible, so the leaf can flex and bend without damaging the delicate cells inside. Veins branch throughout the leaf, delivering water to the photosynthesizing cells and carrying away the sugars they produce. The process of photosynthesis in cherry leaves involves two main stages: the light-dependent reactions and the light-independent reactions, often called the Calvin cycle. In the light-dependent reactions, which occur in the thylakoid membranes inside chloroplasts, light energy is captured by chlorophyll and other pigments and used to split water molecules. This produces oxygen, which is released, and energized electrons, which are used to generate ADP and NADPH, energy-rich molecules. These reactions happen incredibly quickly, with each chlorophyll molecule capturing and processing photons many times per second. The oxygen released by this process is what makes the atmosphere breathable for animals, a gift from plants that sustains nearly all complex life on Earth. The Calvin cycle, which occurs in the stroma of the chloroplast, uses the ADP and NADPH from the light reactions to convert carbon dioxide into sugars. This process is slower and more complex than the light reactions, involving a series of enzyme-catalyzed steps that build up carbon compounds. The key enzyme, rubus-CO, is one of the most abundant proteins on Earth, reflecting its central role in nearly all plant life. This enzyme catalyzes the attachment of carbon dioxide to a 5-carbon sugar, starting the process that eventually produces glucose and other sugars. These sugars are the foundation of the tree's metabolism, providing both energy and the carbon skeletons for building all other organic molecules. Some of the sugar is used immediately to support the leaf's own metabolism. Some is transported to other parts of the tree, and some is converted to starch for storage. Cherry trees, like all plants, must balance their carbon budget, allocating the products of photosynthesis among competing demands. Growth requires carbon for building new tissues. Reproduction requires carbon for flowers and fruits. Storage requires carbon to build reserves for future use. Maintenance requires carbon to repair damaged tissues and replace proteins and other molecules that break down over time. Defense requires carbon to produce compounds that deter herbivores and pathogens. The tree must allocate its finite resources among these competing needs, and the allocation pattern changes with the tree's age, environmental conditions, and reproductive status. A young tree invests heavily in growth, building the structure needed to compete for light and resources. A mature tree invests more in reproduction, having achieved sufficient size to support seed production. A stressed tree may invest more in defense or storage, prioritizing survival over growth or reproduction. The phenology of cherry trees, the timing of seasonal events like flowering and leaf emergence, is a sensitive indicator of climate conditions. Scientists have studied cherry blossom records from Japan spanning more than 1,000 years, making this one of the longest continuous biological data sets in existence. These records show that bloom dates have advanced, occurring earlier in the spring, particularly in recent decades. This shift correlates with rising temperatures, as warmer springs cause trees to accumulate the necessary heat units earlier in the year. While earlier blooming might seem like merely an aesthetic change, it has ecological implications. If cherry trees bloom before their pollinators are active, pollination success may decline. If they leaf out before the peak abundance of spring caterpillars, birds that time their nesting to coincide with caterpillar abundance may find less food for their chicks. The response of cherry trees to climate change involves multiple factors beyond just temperature. Changes in precipitation patterns affect water availability, influencing tree growth and stress levels. Changes in winter cold may affect dormancy satisfaction, potentially leading to irregular or delayed blooming. Increased atmospheric carbon dioxide, the primary driver of climate change, can actually enhance photosynthesis in the short term, as carbon dioxide is a raw material for the process. However, this carbon dioxide fertilization effect is limited by the availability of other resources, particularly nitrogen and water. A tree growing in nutrient-poor soil or experiencing drought may not benefit from higher carbon dioxide levels. Additionally, any benefits of enhanced photosynthesis must be weighed against the stresses imposed by changing temperature and precipitation patterns. Cherry trees also face changing pest and disease pressures as climate shifts. Warmer winters may allow some insect pests to survive in regions where they previously couldn't, or to complete additional generations per year, increasing their population sizes. Pathogens may also expand their ranges or become more virulent under changing conditions. At the same time, the tree's own defense systems may be affected by climate change. Temperature and water stress can influence the production of defensive compounds, potentially making trees more vulnerable to attack. The complex interactions between trees, pests, pathogens, and climate make predictions difficult, but it's clear that the ecosystems surrounding cherry trees are in flux, with relationships that evolved over millennia now changing within decades. Despite these challenges, cherry trees demonstrate remarkable resilience, a capacity to persist and adapt that reflects millions of years of evolution. The genetic diversity within cherry populations provides raw material for adaptation, with some individuals likely possessing traits that make them better suited to future conditions. Natural selection will favor these individuals, potentially shifting the characteristics of populations over time. This evolutionary process happens too slowly to track in real time, but it's occurring nonetheless, a quiet background process that has allowed life to persist through countless environmental changes over Earth's history. Human efforts to conserve genetic diversity in cherry populations through seed banks, botanical gardens, and protected natural areas help ensure that this adaptive potential is preserved. The cultivation of cherry trees by humans has created a parallel process of artificial selection, with people choosing which trees to propagate based on desired characteristics. This has produced the remarkable diversity of ornamental cherry cultivars we see today, each selected for specific traits like flower color, form, blooming time, or growth habit. Some cultivars have been maintained for centuries through careful vegetative propagation, preserving the exact genetic makeup of a single exceptional tree. The famous Sumai Yoshino cherry, which dominates cherry blossom displays in Japan and has been planted widely around the world, is a single clone, meaning every tree is genetically identical. This uniformity creates spectacular synchronized blooming displays, but also means that all these trees share the same vulnerabilities to pests, diseases, and environmental stresses. The practice of grafting, which allows cultivars to be propagated while maintaining their exact characteristics, is an ancient horticultural technique that has been refined over thousands of years. In grafting, a cutting from the desired cultivar, called the scion, is attached to the root system of another tree, called the rootstock. The cambial tissues of scion and rootstock are aligned and held in contact until they grow together, forming a permanent union. The rootstock provides the root system and influences characteristics like tree size and stress tolerance, while the scion provides the above-ground portion and determines flower and fruit characteristics. This allows horticulturists to combine the best traits of different plants, such as pairing a cultivar with exceptional flowers with a rootstock that's disease-resistant or adapted to particular soil conditions. The healing process that occurs at a graft union is a remarkable example of plant regenerative capacity. Initially, the cut surfaces of scion and rootstock are simply held in contact, with no living connection between them. Cells at the cut surfaces respond to the wound by producing callus, a mass of undifferentiated cells that fills the gap between scion and rootstock. Within this callus, some cells differentiate into new vascular tissue, forming xylem and phloem that connect the water and nutrient transport systems of the two partners. This process takes several weeks to months, during which the graft must be protected and kept stable. Once the vascular connection is established, water and nutrients can flow from the rootstock to the scion, and sugars can flow from the scion to the rootstock, supporting both partners. The graft union becomes stronger over time as more tissue forms, eventually becoming nearly as strong as normal wood. The science of plant propagation extends beyond grafting to include other techniques like cuttings, layering, and tissue culture. Cuttings involve removing a piece of stem or root and encouraging it to develop into a complete plant. Cherry trees can be propagated from softwood cuttings taken in early summer, when the new growth is still flexible and green. These cuttings are treated with rooting hormones, usually synthetic auxins that promote root development, and placed in a humid environment with appropriate light and temperature. Under these conditions, cells at the base of the cutting dedifferentiate and form a callus, from which new roots emerge. This process takes advantage of the plant's natural wound response and its capacity to regenerate complete organisms for fragments. Tissue culture, or micropropagation, takes plant regenerative capacity to an extreme. In this technique, tiny pieces of plant tissue, sometimes just a few cells, are placed in a sterile nutrient medium containing sugars, minerals, vitamins, and hormones. Under carefully controlled conditions, these cells can be induced to form callus, which can then be stimulated to produce shoots and roots, developing into complete plantlets. This technique allows a single plant to be multiplied into thousands or millions of identical copies, all in a relatively small space. It's particularly useful for rare or valuable cultivars, for plants that are difficult to propagate by other methods, or for producing disease-free stock, since the sterile conditions prevent pathogen transmission. The plantlets produced through tissue culture are genetically identical to the parent plant and to each other, creating clonal populations. The cellular mechanisms underlying plant regeneration are areas of active research. Unlike animal cells, which generally become committed to specific fates and lose the ability to change, many plant cells retain remarkable plasticity. A differentiated cell in a stem or leaf, under the right hormonal signals, can dedifferentiate, essentially reverting to a stem cell-like state, and then redifferentiate into a completely different cell type. This plasticity is controlled by complex networks of genes and hormones, with auxins and cytokinins playing particularly important roles. The ratio of these hormones influences whether cells form roots, shoots, or callus. High auxin relative to cytokinin promotes root formation. High cytokinin relative to auxin promotes shoot formation, and intermediate ratios promote callus formation. By manipulating hormone levels, researchers and horticulturists can guide plant development, coaxing cells to form desired structures. This regenerative capacity has implications beyond horticulture. It's a fundamental aspect of how plants grow and respond to their environment. When a branch breaks, the tree can produce new shoots from dormant buds below the break. When roots are damaged, new roots can form from the remaining tissue. This modular growth pattern, where the plant consists of repeated units that can be added or removed without killing the organism, is fundamentally different from animal development. Animals generally have a determinate body plan, with a fixed number of organs in fixed positions. Plants have an indeterminate body plan, continuously producing new organs throughout their lives. A cherry tree doesn't have a predetermined number of branches or leaves. It keeps making more as long as it lives and conditions permit. This continuous growth means that a tree is, in a sense, a colony of modules, each capable of some degree of independent function. A branch with leaves can photosynthesize and produce sugars, some of which support that branch's own growth, and some of which are shared with the rest of the tree. Roots in a resource-rich patch of soil can proliferate, producing more fine roots to exploit that patch, while roots in poor soil may grow more slowly or die back. The tree integrates the activities of all these modules, sharing resources through the vascular system and coordinating growth through hormonal signals. but there's also a degree of local autonomy. This modular organization contributes to the tree's resilience. If one branch dies, others continue functioning, and new branches can grow to replace the lost one. The longevity of cherry trees varies widely among species and growing conditions. Wild cherry trees in favorable environments can live for a century or more, slowly growing into substantial trees. The oldest known cherry trees are several hundred years old. Their gnarled trunks and spreading branches testament to their endurance. Ornamental cherry cultivars, particularly those grown in urban environments, often have shorter lifespans, perhaps 50 to 80 years, due to the stresses of urban conditions and sometimes to characteristics of the cultivar itself. Some cultivars with particularly showy flowers or unusual growth forms may be less vigorous than wild types, a trade-off between aesthetic appeal and longevity. Yet, even a few decades is a substantial span from a human perspective, long enough for a tree to be enjoyed by multiple generations. The cherry blossom tree stands as a testament to cycles that extend beyond any single bloom, beyond any single season. The biological mechanisms we have explored tonight are not isolated processes, but threads in a continuous tapestry of renewal that has unfolded for millions of years. The Prunus genus evolved during the Paleogene period, roughly 60 million years ago, when the world was still recovering from the great extinction that ended the age of dinosaurs. In those ancient forests, the ancestors of cherry trees began developing the strategies we see today, the capacity to synchronize blooming, to attract pollinators, to survive cold winters and emerge transformed each spring. The fossil record preserves fragments of this evolutionary journey. Paleobotanists have discovered fossilized prunus leaves and seeds in rock formations across the northern hemisphere, evidence that these trees spread gradually from their origins in Central Asia. The shapes of ancient leaves reveal adaptations to changing climates, shifting rainfall patterns, and the gradual cooling that occurred throughout the Cenozoic era. Each fossil represents a moment frozen in time, a single leaf that fell perhaps 30 million years ago, now pressed in a stone, its veins still visible, its serrated edges still distinct. The evolutionary success of cherry trees lies partly in their flexibility. Unlike some plant lineages that became highly specialized and vulnerable to environmental change, the Prunus genus maintained a remarkable adaptability. Different species evolved to thrive in diverse habitats, from the cold mountains of Japan to the temperate valleys of Europe, from the Mediterranean coast to the high plateaus of the Himalayas. This geographic diversity meant that when climates shifted, when ice ages advanced and retreated, cherry trees could migrate, could find new territories, could survive in refugia, and then expand again when conditions improved. The relationship between cherry trees and their pollinators has also evolved over millions of years. The earliest flowering plants relied on wind and water for pollination, but the evolution of flowers created opportunities for partnerships with insects. Bees, butterflies, and other pollinators evolved alongside flowering plants in a process called coevolution. As flowers developed more attractive colors and fragrances, pollinators evolved better vision and scent detection. As pollinators became more efficient, flowers developed more specialized structures to guide them. This mutual adaptation created the intricate relationships we see today, where a bee can navigate directly to nectar, where a flower's shape perfectly matches a pollinator's body. The cherry blossom's brief lifespan is not a flaw, but an evolutionary strategy. By blooming early and quickly, cherry trees take advantage of a window when competition for pollinators is relatively low. Many other trees have not yet flowered. Many other nectar sources are not yet available. So bees and other insects concentrate their attention on the abundant cherry blossoms. This timing also means that seeds develop and disperse before the hottest, driest part of summer, giving seedlings time to establish roots before their first winter. The strategy of ephemerality, of brief but intense flowering, has proven successful across millions of years. Within each cherry tree, genetic information accumulated over countless generations guides every developmental process. The tree's DNA contains roughly 340 million base pairs, sequences of nucleotides that encode instructions for building proteins, regulating genes, and responding to environmental signals. This genome is not static, but dynamic, shaped by mutation, selection, and genetic drift. Each tree carries slight variations, unique combinations of genes that may affect bloom timing, petal color, disease resistance, or drought tolerance. This genetic diversity is the raw material for future evolution, the reservoir of possibilities that allows cherry trees to adapt to changing conditions. The process of gene expression in cherry trees responds to seasonal cues with remarkable precision. Certain genes activate only when temperatures drop below specific thresholds, triggering the production of cold protective proteins. Other genes activate when day length increases in spring, initiating the cascade of hormonal changes that lead to bud break. Still other genes respond to stress, to drought, to pathogen attack, producing defensive compounds or signaling molecules. This complex regulatory network allows a single genome to produce different responses depending on the environment, a flexibility that is essential for survival. The study of cherry tree genetics has revealed surprising connections to other plant families. Genes that control flower development in cherry trees are similar to genes found in completely unrelated plants, evidence of deep evolutionary conservation. These master regulatory genes, including those in the MADS-box family, are ancient, dating back hundreds of millions of years to the common ancestors of all flowering plants. The fact that such similar genes control flower development in roses, orchids, and cherry trees suggests that the basic toolkit for building flowers evolved early and has been modified and refined in different lineages. Epigenetic mechanisms add another layer of complexity to cherry tree biology. Beyond the DNA sequence itself, chemical modifications to DNA and histone proteins can affect gene expression without changing the underlying genetic code. These epigenetic marks can be influenced by environmental conditions and, in some cases, can be passed from one generation to the next. A tree that experiences severe drought might develop epigenetic modifications that make its offspring slightly more drought tolerant, a form of inheritance that complements traditional genetic variation. This epigenetic memory allows trees to respond not just to current conditions, but to the environmental history experienced by their ancestors. The cherry tree's vascular system, which we touched upon earlier, represents an engineering marvel refined over millions of years. The xylem tissue that transports water from roots to leaves must overcome significant physical challenges. Water must be lifted against gravity, sometimes to heights of 10 or 15 meters in large trees. This lifting occurs through a combination of root pressure, capillary action, and transpirational pull. As water evaporates from leaf surfaces, it creates negative pressure that pulls water upward through the xylem vessels, like sucking liquid through a straw. The cohesive properties of water molecules, which tend to stick together, allow this column of water to remain continuous even under tension. The xylem vessels themselves are dead cells at maturity, their living contents having broken down to leave hollow tubes. The walls of these tubes are reinforced with lignin, a complex polymer that provides structural support and prevents collapse under negative pressure. Different patterns of lignin deposition create different vessel types, some adapted for maximum water transport, others for greater safety against cavitation. The evolution of increasingly efficient xylem structures was crucial to the success of trees, allowing them to grow taller and access more light while maintaining water transport from deep roots. The phloem tissue, which transports sugars and other organic compounds, faces different challenges. Unlike xylem, phloem cells remain alive at maturity, though they lose their nuclei and many other organelles. The sieve tube elements that form the main transport pathway are connected by sieve plates, specialized cell walls with large pores that allow fluid to flow from cell to cell. Companion cells adjacent to sieve tube elements provide metabolic support, supplying energy and proteins needed to maintain transport. The pressure flow mechanism that drives phloem transport relies on osmotic gradients, with sugars loaded into phloem at source tissues like leaves and unloaded at sink tissues like roots, flowers, and developing fruits. The allocation of resources within a cherry tree involves complex decision-making at the physiological level. The tree must balance investments in different structures and functions, allocating carbon and nutrients to roots, trunk, branches, leaves, flowers, and fruits. These allocation patterns change with the seasons and with the tree's developmental stage. A young tree invests heavily in root and shoot growth, establishing its structure. A mature tree allocates more resources to reproduction, producing abundant flowers and fruits. A stressed tree might prioritize survival, reducing growth and reproduction to maintain essential functions. Hormonal signals coordinate these allocation decisions. Cytokinins produced in roots promote shoot growth. Auxins produced in shoot tips promote root growth and inhibit lateral bud development. Gibberellins promote stem elongation and fruit development. Abscisic acid helps the tree cope with stress, closing stomata to conserve water and promoting dormancy. Ethylene triggers fruit ripening and leaf senescence. These hormones interact in complex networks, with the effect of one hormone often depending on the presence and concentration of others. The tree's physiological state emerges from these interactions, a dynamic equilibrium constantly adjusting to internal and external conditions. The cherry tree's immune system, though different from animal immunity, is equally sophisticated. Plants cannot produce antibodies or mobilize immune cells, but they have evolved multiple layers of defense against pathogens. The first layer is physical, the barriers of bark, cuticle, and cell walls that prevent pathogen entry. The second layer is chemical, the production of antimicrobial compounds that inhibit pathogen growth. The third layer is systemic, a signaling network that allows the tree to recognize pathogen attack and mount a coordinated defense response. When a pathogen breaches physical barriers and enters plant tissue, the tree cells detect molecular patterns associated with pathogens, molecules like chitin from fungal cell walls or flagellin from bacterial flagella. This pattern recognition triggers a cascade of defensive responses. The plant produces reactive oxygen species that damage pathogen cells. It synthesizes antimicrobial proteins and secondary metabolites. It reinforces cell walls at the infection site. In some cases, infected cells undergo programmed cell death, sacrificing themselves to prevent pathogen spread. This hypersensitive response creates a zone of dead tissue around the infection site, effectively walling off the pathogen. The tree can also develop systemic acquired resistance, a form of immunological memory. After successfully defending against a pathogen, the tree becomes more resistant to subsequent infections, not just at the original infection site, but throughout the entire plant. This enhanced resistance involves the production of signaling molecules like salicylic acid, which travel through the vascular system and prime distant tissues for faster, stronger defensive responses. Though different in mechanism from animal immunity, this system achieves a similar result, allowing the tree to remember past threats and respond more effectively to future challenges. The cherry tree's relationship with beneficial microorganisms extends beyond mycorrhizal fungi. The phyllosphere, the aerial surface of leaves, hosts diverse communities of bacteria, yeasts, and other microorganisms. Some of these microbes, are simply opportunistic colonizers, but others provide benefits to the tree. Certain bacteria produce antibiotics that suppress pathogenic fungi. Others fix atmospheric nitrogen, though in much smaller quantities than the nitrogen-fixing bacteria associated with legumes. Still others produce plant growth hormones that can enhance leaf development. The tree, in turn, secretes sugars and other organic compounds from its leaf surfaces, providing food for these microbial communities. The rhizosphere, the zone of soil immediately surrounding roots, is even more densely populated with microorganisms. A single gram of rhizosphere soil can contain billions of bacterial cells representing thousands of species. These microbes are attracted by root exudates, the complex mixture of sugars, amino acids, organic acids, and other compounds that roots release into the soil. Some rhizosphere bacteria solubilize minerals, making phosphorus and other nutrients more available to the tree. Others produce siderophores, molecules that bind iron and make it accessible to plant roots. Still others induce systemic resistance, priming the tree's immune system without causing disease themselves. The concept of the holobiont, the tree plus all its associated microorganisms functioning as a single ecological unit, has gained increasing recognition in recent years. The cherry tree does not exist in isolation, but is part of a complex community where boundaries between individual organisms blur. The tree's health and function depend not just on its own genes, but on the collective genetic capacity of its entire microbiome. This perspective shifts our understanding of what constitutes an individual organism, suggesting that life exists not in isolation, but in relationship, in networks of interdependence that span scales from molecular to ecological. The cherry blossom's cultural significance across East Asia reflects not just aesthetic appreciation, but deep philosophical understanding of impermanence and renewal. The Japanese concept of mono no aware, the poignant awareness of the transience of things, finds perfect expression in cherry blossoms. The flower's brief existence reminds us that beauty need not be eternal to be meaningful, that there is profound value in moments that pass. This recognition of impermanence is not melancholic but liberating, an acceptance that change is the fundamental nature of existence. In Chinese culture, cherry blossoms symbolize feminine beauty and the power of gentleness. Classical poetry often uses cherry blossoms as metaphors for grace, elegance, and the strength that comes from flexibility rather than rigidity. The blossoms that bend in the wind but do not break, that fall gracefully rather than clinging desperately to branches, embody a wisdom about how to move through life with resilience and dignity. These cultural meanings, developed over centuries, reflect careful observation of natural phenomena and thoughtful reflection on their implications for human existence. The practice of contemplating cherry blossoms, of sitting quietly beneath blooming trees, and simply being present with their beauty, can be understood through the lens of modern psychology and neuroscience. When we engage in such contemplation, our minds shift from the default mode of planning, worrying, and ruminating to a more present-focused awareness. This shift activates different neural networks, reducing activity in regions associated with self-referential thinking and increasing activity in regions associated with sensory processing and attention. The result is a state of calm alertness, a reduction in stress hormones, and an increased sense of connection to the present moment. The aesthetic response to cherry blossoms involves multiple brain regions working in concert. The visual cortex processes the colors, shapes, and patterns of the flowers. The limbic system, particularly the amygdala and nucleus accumbens, generates emotional responses and feelings of pleasure. The prefrontal cortex integrates sensory information with memories and cultural meanings, creating a rich, multidimensional experience. The release of neurotransmitters like dopamine and serotonin contributes to feelings of happiness and well-being. This neurological response to natural beauty may have evolutionary roots, with preferences for certain landscapes and natural features having provided survival advantages to our ancestors. The phenomenon of forest bathing, or Shinrin-yoku, developed in Japan in the 1980s, formalizes the health benefits of spending time in nature. Research has shown that walking in forests, particularly during cherry blossom season, reduces cortisol levels, lowers blood pressure, improves immune function, and enhances mood. These benefits appear to result from multiple factors: the reduction in urban noise and air pollution, the engagement of attention by natural stimuli, the presence of phytoncides and other volatile organic compounds released by trees, and perhaps the activation of innate human responses to natural environments. The color pink itself, so prominent in cherry blossoms, has psychological effects that researchers have documented. Pink is generally perceived as calming and soothing, associated with gentleness, nurturing, and tranquility. Exposure to pink environments can reduce aggressive behavior and promote feelings of relaxation. The particular shades of pink found in cherry blossoms, often described as soft, delicate, or pastel, may be especially effective at inducing calm states. This response likely combines learned cultural associations with innate perceptual preferences, creating a powerful emotional resonance. The fragrance of cherry blossoms, subtle though it is, contributes to their psychological impact. As we discussed earlier, the volatile compounds that create this fragrance include molecules that interact with olfactory receptors in the nose, sending signals to the olfactory bulb and then to the limbic system. Because the olfactory system connects directly to brain regions involved in emotion and memory, scents can trigger powerful emotional responses and vivid memories. The scent of cherry blossoms might evoke memories of past springs, of moments of happiness or peace, creating a sense of continuity across time. The temporal aspect of cherry blossom viewing, the awareness that the flowers will soon fall, adds psychological depth to the experience. This awareness of transience can enhance appreciation, making each moment feel more precious. Psychological research on savoring, the deliberate attention to positive experiences, suggests that awareness of an experience's temporary nature increases its emotional impact. When we know that something beautiful will not last, we attend to it more fully. We notice details we might otherwise overlook. We feel more grateful for its presence. The social dimension of hanami, the tradition of gathering with friends and family to view cherry blossoms, provides additional psychological benefits. Social connection is fundamental to human well-being, and shared positive experiences strengthen social bonds. The ritual of hanami creates a culturally sanctioned opportunity for people to slow down, to spend unhurried time together, to share food and conversation in a beautiful setting. These gatherings reinforce community ties and create shared memories, contributing to both individual and collective well-being. The cherry tree's response to climate change, which we have explored throughout this journey, represents a real-time experiment in evolution and adaptation. As temperatures rise and precipitation patterns shift, cherry trees face novel environmental conditions. Some populations may adapt through natural selection, with individuals that happen to carry genes for later blooming or greater drought tolerance, leaving more offspring. Other populations may adapt through phenotypic plasticity, adjusting their development in response to environmental cues without genetic change. Still, other populations may decline or disappear, unable to adapt quickly enough to rapidly changing conditions. The speed of current climate change presents unprecedented challenges. Evolutionary adaptation typically occurs over many generations, allowing gradual accumulation of beneficial mutations. Rapid climate change compresses the timescale, potentially exceeding the rate at which natural selection can operate. Phenotypic plasticity can provide some buffer, allowing trees to adjust their physiology and development, but plasticity has limits. If environmental conditions shift beyond the range that a tree's existing genetic and physiological systems can accommodate, decline becomes inevitable unless human intervention provides assistance. Assisted migration, the deliberate movement of tree populations to more suitable climates, represents one potential conservation strategy. If cherry trees in a particular region are declining because local conditions have become too warm or too dry, moving seeds or saplings to cooler or wetter areas might allow the population to survive. This approach raises complex ecological and ethical questions. Will moved trees disrupt existing ecosystems? Will they become invasive? Do humans have the right or responsibility to intervene in natural processes? These questions have no simple answers, but they reflect the reality that human actions have already profoundly altered the planet, and passive observation may not be sufficient to preserve biodiversity. Ex situ conservation, the preservation of genetic diversity in botanical gardens, seed banks, and arboreta, provides another safety net. By maintaining living collections and frozen seed samples of diverse cherry tree varieties, conservationists ensure that genetic diversity is not lost even if wild populations decline. These collections serve as genetic libraries, preserving options for future restoration efforts. They also provide material for research, allowing scientists to study how different genetic variants respond to various environmental conditions, knowledge that can inform both conservation and breeding efforts. The intersection of traditional knowledge and modern science offers valuable insights for cherry tree conservation. Indigenous and local communities often possess detailed knowledge of tree behavior, accumulated through generations of observation. This knowledge can complement scientific research, revealing patterns and relationships that might not be apparent from short-term studies. Collaborative approaches that respect and integrate different knowledge systems may prove most effective for understanding and protecting cherry trees in a changing world. The cherry blossom's role as a phenological indicator, a marker of seasonal change, makes it valuable for climate research. Long-term records of bloom dates provide data about temperature trends and climate variability. As these records extend further into the future, they will help scientists understand the pace and pattern of climate change, test climate models, and predict future conditions. In this way, cherry trees contribute not just beauty but knowledge, helping humanity understand the changes we are causing and the adaptations we must make. The economic value of cherry blossoms, particularly in Japan, demonstrates how nature can support human livelihoods while remaining intact. The cherry blossom season generates billions of dollars in tourism revenue, supporting restaurants, hotels, transportation services, and local businesses. This economic value creates incentives for conservation, as communities recognize that protecting cherry trees protects their economic well-being. The challenge lies in managing tourism sustainably, ensuring that the influx of visitors does not damage the trees or the ecosystems that support them. The art inspired by cherry blossoms spans centuries and continents, from ancient Japanese woodblock prints to contemporary photography, painting, poetry, and music. This artistic engagement with cherry blossoms reflects and reinforces their cultural significance, creating feedback loops where art shapes how people perceive cherry blossoms, and cherry blossoms inspire new art. The aesthetic traditions that have developed around cherry blossoms influence how people experience them, guiding attention to particular qualities, suggesting meanings and associations, creating frameworks for appreciation. The symbolism of renewal embodied by cherry blossoms extends beyond the individual tree to broader cycles of regeneration. Each spring, as cherry trees bloom, they participate in the planet's seasonal rhythm, the greening of the northern hemisphere, the return of warmth and growth after winter's dormancy. This planetary rhythm, driven by Earth's axial tilt and orbital motion, has structured life's evolution for billions of years. The capacity to respond to seasonal change, to synchronize life cycles with predictable environmental patterns, has been crucial to the success of countless species. The cherry tree's annual cycle of growth, reproduction, and dormancy mirrors cycles at every scale of existence. Cells cycle through phases of growth and division. Ecosystems cycle nutrients through food webs. The planet cycles carbon between atmosphere, ocean, and land. The universe cycles matter and energy through stars, which forge heavy elements and scatter them through space when they die, seeding new generations of stars and planets. These nested cycles create the conditions for complexity, for the emergence of patterns and structures, for life itself. The concept of renewal implicit in cherry blossoms suggests that endings are also beginnings, that death feeds life, that loss makes space for growth. When cherry blossoms fall, they do not simply disappear, but transform, becoming food for decomposers, nutrients for soil, energy for new growth. This transformation is not metaphorical, but literal, a physical process of breaking down complex molecules and reassembling them into new forms. The carbon atoms in a fallen petal might become part of a fungal hypha, then part of the soil organic matter, then taken up by a root, then incorporated into a new leaf. Matter cycles, energy flows, and patterns persist even as their material substrate constantly changes. The persistence of cherry trees through millions of years of environmental change testifies to life's resilience and adaptability. The lineage has survived ice ages and warm periods, volcanic eruptions, and asteroid impacts, the rise and fall of countless other species. This survival reflects not invulnerability, but flexibility, the capacity to change, to adapt, to find ways to persist even as conditions shift. The cherry trees blooming today carry within them the accumulated wisdom of millions of years of evolution, solutions to problems of survival and reproduction tested and refined across deep time. As you rest tonight, you might hold in your mind the image of a cherry blossom, its delicate petals backlit by spring's sun, its subtle fragrance carried on a gentle breeze. You might consider the complexity hidden within that simple beauty, the molecular machinery, the evolutionary history, the ecological relationships, the cultural meanings. You might reflect on how something so ephemeral can be so meaningful, how a flower that lasts only days can symbolize enduring truths about existence, change, and renewal. The cherry blossom teaches that beauty does not require permanence, that there is profound value in the fleeting and temporary. It teaches that endings are natural and necessary, that letting go allows new beginnings. It teaches that we are part of larger cycles, connected to patterns that extend beyond our individual lives, participating in rhythms that have pulsed through the planet for eons. These lessons, encoded in the biology and symbolism of cherry blossoms, offer comfort and perspective, reminding us of our place in the vast, interconnected web of existence. The science we have explored tonight reveals cherry blossoms as far more than decorative objects. They are products of evolutionary processes spanning millions of years, participants in complex ecological networks, indicators of environmental change, sources of economic value, inspirations for art and philosophy, and teachers of wisdom about impermanence and renewal. Understanding the science deepens rather than diminishes their beauty, adding layers of meaning and connection, revealing the cherry blossom as a point where multiple scales and dimensions of reality intersect. Your mind may be growing quiet now, thoughts slowing like petals drifting down. The facts and details we have explored can settle into your memory, patterns and images and ideas that might surface later, in dreams or waking moments. bringing with them a sense of calm and connection. There is no need to remember everything, no need to hold on. Like cherry blossoms themselves, thoughts can come and go, each one valuable in its moment, each one part of a larger pattern. If you are still awake, still listening, know that there are countless other stories waiting, other journeys through science and nature that can accompany you toward rest. The universe is vast and full of wonders, from the smallest quantum fluctuations to the largest cosmic structures, from the deepest ocean trenches to the highest mountain peaks, from the simplest bacteria to the most complex ecosystems. Each of these realms offers opportunities for exploration, for learning, for the quiet pleasure of understanding how things work and why they matter. But for now, for tonight, the cherry blossoms have shared their story. They have opened their petals to reveal the hidden complexity within, the science and history and meaning woven together in each flower. They have reminded us that renewal is possible, that spring follows winter, that beauty emerges from the simplest elements, carbon and hydrogen, oxygen and nitrogen, arranged in particular patterns, animated by energy from the sun, shaped by evolution, experienced by consciousness. The cherry tree stands in the darkness now, its blossoms closed or perhaps already fallen, its branches swaying gently in the night breeze. Beneath the soil, its roots extend through the earth, connected to fungi, to other trees, to the vast underground network of life. Within its cells, genes switch on and off, proteins fold and unfold, molecules move and react. All the quiet machinery of life continuing its work. The tree does not sleep, not in the way we do, but it rests in its own fashion, conserving energy, maintaining essential functions, waiting for the sun to rise again. And you too can rest now. You can let your breathing deepen and slow. You can allow your muscles to relax, your thoughts to drift. You can trust that the world will continue its cycles, that spring will come again, that cherry blossoms will bloom, that renewal is woven into the fabric of existence. You are safe. You are part of these larger patterns. You are connected to everything we have explored tonight, to the trees and the soil, to the pollinators and the fungi, to the long evolutionary journey that has brought us all to this moment. The story of cherry blossoms continues beyond this telling, beyond this night. Tomorrow, somewhere in the world, cherry trees will be blooming. People will gather beneath them, will pause in their busy lives to look up at the flowers, will feel something shift inside themselves, some recognition of beauty and transience and connection. The trees will photosynthesize and transpire, will interact with their mycorrhizal partners, will provide habitat for insects and birds. The cycle will continue, as it has for millions of years, as it will for years to come. Rest now, knowing that you have traveled through deep time and intricate space, through the biology and ecology and culture of cherry blossoms. Rest knowing that renewal is always possible, that spring follows winter, that beauty emerges even from bare branches. Rest in the knowledge that you are part of something larger, connected to patterns and cycles that extend beyond any individual life, participating in the great dance of existence that includes cherry trees and human beings, microbes and galaxies, matter and energy, time and space. The cherry blossoms have shared their wisdom. Now they release their petals to the wind, returning their substance to the earth, completing their cycle, making space for what comes next. And you, too, can release, can let go, can allow yourself to drift into sleep, trusting in the renewal that morning brings, knowing that you are held by the same forces that guide the cherry tree through its seasons, the same patterns that have sustained life on this planet for billions of years. Sleep well, rest deeply, and may your dreams be gentle as falling petals, soft as spring rain, peaceful as a cherry tree standing silent in the moonlight. Its work complete, its beauty offered freely to the world, its promise of renewal echoing through the quiet night.